to celebrate and strengthen our community. I sincerely thank everyone who dedicated their time and generosity to make this event possible. I am truly grateful for the opportunity to kick off this meeting in Buenos Aires, a city renowned for its hospitality, creativity, and enduring spirit of collaboration. This year's theme, From Data to Discovery, Strengthening Latin America's Bioimaging Ecosystem, captures our shared goal to turn challenges into opportunities, build meaningful partnerships, and drive innovation that will shape the future of our field. Over the next few days, I encourage everyone to approach this with curiosity, generosity, and a focused mindset. Let's take this opportunity to share knowledge, spark new ideas, and foster lasting collaborations, building a more open, connected, and impactful scientific ecosystem. Together, we can push the boundaries of bioimaging, positioning Latin American researchers as global leaders in scientific progress. Welcome to LABI 2025. May this meeting be both memorable and transformative for everyone. Thank you.
Okay, now we go with Kildan Miranda from Universidade Federal do Rio de Janeiro, Chair of LABI.
Yes. Okay. Okay, thank you. Thank you. So good morning, everyone. So first of all, welcome to Buenos Aires and welcome to LABI Meeting 2025. Not Rio, as Oliver already mentioned. So my name is Kildan Miranda. I'm the chair of Latin America Bioimaging. I'm a professor at the Federal University of Rio de Janeiro. And it is with immense pride and sense of accomplishment that I see this room full of friends and people that we know a lot that dedicate themselves to the bioimaging community. Just a little over 3 years ago, we started as 70 people in 7 countries, and the community evolved very, very fast. So we grew from over 70 people to 800 now, as I'm going to show the numbers here. It's really very good to see that we were able to grow very fast this community that is very committed to push forward bioimaging capacity in our region. So this is just not a growth, but it's a testament of our dedication and success of our shared vision that has come into practice. So thanks to all of you and to the LABI programs that we were able to put together, we were able to grow this community. And we have successfully transitioned from an informal community to a recognized regional platform. And now it's time to reassess our goals and define the next era of our collective effort and our impact in the region. So, having said that, as Leah mentioned, so we know that the world is becoming a very strange place with wars, surveilling drones, funders shifting their priorities, governments shifting also their vision of science. But as Leah mentioned, we didn't chat before. We know that our community is very resilient, and we know how to build, to open dialogues with the diverse sectors, even when the winds are not in our favor, right? As is the case of some countries now, within or outside Latin America. This is happening everywhere. And bioimaging is a common language that we share. And it's a language that unites us, and it's a language of discovery, shared vision for a better human endeavor. So I hope that these next days we use this unity and this shared scientific passion as our resilient fuel to drive and to find creative solutions for this situation that we live. So welcome again. I wish you an excellent meeting, and I hope that we'll have fruitful discussions over the next days. Thank you very much.
Okay, so moving forward, in these numbers and this community that Hilary was mentioning and showing, I want to show only which are the main activities in which we were focusing. I'm not going to get deep on this. We are going to have more information during the whole meeting and during the 2 days. But in big words, we get together the specialists and our community into the working groups, working in regional strategies, for example, in training and education, you're going to see a regional curricula in fundamentals of microscopy. Also with the data science for bioimaging working group, working on making bioimaging data fair and also ready to AI and things like that. So we are going to see that later. We also get more impact into collaborative projects, and we have a strong network, and we have been supporting the exchange of knowledge and also strengthening collaborations on the fellowships. We have nowadays 73 fellows that have been supported by LABI in the last 3 years from 9 countries. And we get together into these annual meetings, trying to mainly think the strategic conversation, as Hilary mentioned, for the region in terms of bioimaging, access to technology, and training. So, going specifically to the annual meetings, that is what we are doing here. And giving some perspective, in 2022, we met in Montevideo, Uruguay, 50 persons from 11 countries there in Montevideo. Really a nice meeting. Then we traveled to Mexico, Morelos, Cuernavaca, with almost 90 participants from 10 countries. Then we moved, to Rio at this time, and we grow also with the local community into 110 participants, and also increasing in the participation of the level of countries. And nowadays, on this meeting, on 130 participants from 11 countries, there are 15 countries from Latin America. And something important is not only the numbers of growing, because we limit the capacity of this type of meeting. We cannot have a meeting more than 120, 130 persons, because the level of interaction of our meetings. So we are really happy, and we receive, of course, a lot of applications to participate in the meeting, but also a lot of applications to the travel grants. In this case, to the travel grants for this meeting, we received more than 100 applications from 13 countries, and many of you are here, and we really appreciate that effort. And we really thank that you are here. So, the main goals of this meeting, as Hilary and Lee already mentioned, as a community, but specifically on this space, to identify challenges inside, but also between the regions. So we have community partners from other regions, from Africa, from Bioimaging North America, from Africa, and promote the collaboration and expand our network. So you are part of this community, you are this community, so that is something extremely important for us, that you can feel that, and integrate into the whole global ecosystem. So at the end, this can really grow the strategic convergence in our region. So for this, it's for us extremely important that this environment that we're creating is something of shared responsibility. So we have a code of conduct. You already received that, you really consent that, and it's extremely important for us. You can also access it there. And we expect that all participants really contribute to this environment on the exchange, collaboration, and also growing this space. The main topics. During these 2 days, we are going to go through technology in action in the first day, to data science and infrastructure in data in the second day. On the third day, on Wednesday, we're going to have the facility visits and the outreach activities. Then, in each of the day, we're going to get more deep on it. And we were also, with the satellite event that we have the past week on the Formisur, this super resolution microscopy training at Sibion. And on Wednesday, we'll start the array tomography workshop. Here will be Mariano here from this community. So thank you very much for all this meeting to the program organizing committee, also in the communications and logistics, Ana Clara and Silvina, that were essential for this. All the local committee, that is also something extremely valuable for us when we do our meetings in different countries. So this is something extremely valuable for the community. And the coordinators of the working groups that during the whole year are working also this with the rest of the community that is participating on these working groups. And also, thank you to the partner organizations. These organizations are the ones that are going to be working on different spaces during this meeting, trying to see this strategic convergence between regions and also inside our region. And thanks to the supporters, in this case, Chance Zuckerberg Initiative, for this meeting, all the sponsors, but more especially to the diamond sponsors, Helena Bio, the platinum sponsors, Science and ABEF, and the gold sponsors, Leica, Thermo Fisher, and Tisnotics. But thank you all sponsors for being here. And also, they are all invited to participate as one more community member. So let's continue now, and we go with the keynote presentation by Fernando. Thank you very much, and welcome.
It's a great honor to welcome our keynote speaker, Professor Dr. Fernando Stefani. Dr. Stefani is a principal researcher at the National Scientific and Technical Research Council, CONICET, a full professor of experimental physics in the physics department at Exactas, and the director of the Center for Bioscience Research at CONICET. Internationally recognized for his pioneering work in nanophotonics and super-resolution microscopy, Dr. Stefani earned his doctorate with highest honors in Germany and completed postdoctoral research at leading European institutions. Upon returning to Argentina, he established a world-class laboratory at the University of Buenos Aires and CONICET. Dr. Stefani's research has advanced fluorescence imaging and nanotechnology, resulting in worldwide recognition for his breakthrough visualization methods. His leadership has transformed his laboratory into a regional hub of innovation, inspiring and supporting the careers of over 30 young scientists, many of whom now work in academia and industry worldwide. With over 90 publications in leading journals such as Nature, Science, and ACS Nano, and a series of prestigious awards, including the Otto Hahn Medal, the Georg Foster Research Award from the Alexander von Humboldt Foundation, the Hussey Prize in Mathematics, Physics, Astronomy, and Computation, and the Konex Prize. Dr. Stefani ranks among the most influential scientists in Argentina over the past decade. Beyond his research, Dr. Stefani is a dedicated mentor, a strategic advisor to scientific institutions, and a passionate advocate for advancing science in Latin America. Please join me in welcoming Dr. Stefani, whose vision and achievements continue to shape the future of nanoscience and bioimaging.
Okay, wow, Leah, that's a bit too much. Thank you very much. Good morning, everybody. So, I'm gonna use the few minutes I have this morning to talk to you about technology applied for bioimaging, and particularly to bringing fluorescence microscopy down to molecular level as much as possible. So before I start, just a few words about where we work. It's at the Center for Bio-Nanoscience Research. It's not far from here in a new complex that's called the Polo Científico Tecnológico in Palermo. That's a nice building. We are somewhere, maybe I can use the laser pointer. So we are somewhere in the first and second floor here in this building. It's a relatively small research institute, but we tackle interdisciplinary research among chemists, biologists, and physicists. Here you see a few snapshots of my group over the years. The work I will show you today has involved Francisco Balzarotti, Luciano Mazzullo, Martín Bordenave, Federico Barabás, Cecilia Sasa, Lucia López, Mariano Varela, let me see, Florencia Adorna, Florencia Choque here, Santiago Sosa, and Gonzalo Escalante. I hope I'm not forgetting anyone. So, we are a small group, but we are lucky to have a strong network of collaborators, both in Argentina and around the world. And some of the experiments I will show you today were done with the great help of Stefan Hell and Tom Jovin in Göttingen, Philipp Tinefeld from LMU Munich, Alfredo Cáceres from Córdoba, Sabrina Simoncelli from London, Guillermo Acuña from Fribourg, Switzerland, Oscar Campetela and Juan Mucci from UNSAM, Buenos Aires, and Fernando Goldbaum from Fundación Instituto Leloir in Buenos Aires, and Marcelo Martí and Matthias Blaustein in University of Buenos Aires. So let's get started. But just before, I'm quite sorry that the first paper I will talk about is this one that just appeared in Nature a few months ago. And I will not go into details and be reiterative about the difficulties we are going through in Argentina, but just believe me, it's really serious. Nevertheless, we continue working and we have exciting projects, and I will show you some of them. So let's get started. To this community is, I guess, totally clear that fluorescence microscopy is one of the preferred tools to visualize soft matter and particularly biological matter. That's because fluorescence microscopy gathers a set of features that are quite unique. So it gives us like a very high sensitivity and specificity. We can even detect single molecules. It's non-invasive, so we use basically visible light, and it can work at physiological conditions. That means room temperature, liquid environments. And it also allows us to do 3D imaging. So due to this, fluorescence microscopy is really, really a powerful bioimaging tool. But it has a limitation, as you all know. It's resolution, limited by the wavelength of light. So what you see here is a baby neuron imaged with fluorescence microscopy, and it's, as you can see, it's great to see the morphology of the cell, to see how proteins are organized in different domains of the cell. But if we want to look at this with more detail, like zooming in on this growth cone, we see that the image gets blurred. And that's not because we did anything wrong, that's just because we are reaching the resolution limit of light microscopy, which is about a couple of 100 nanometers. So here is just a few slides I'm going to use to explain how bad it is. So imagine a molecule. That's one of the typical fluorophore. It has a size of about 1 nanometer. If we take an image of a molecule with our fluorescence microscope, we get a signal that looks like this. It's about 200 nanometers. So you see, we start with an object 1 nanometer in size, and we end up with a signal that is 2 nanometers in diameter. So in area it's 40,000, and in 3D the zeros continue to increase. So it's really a lot what we are missing. I typically use this analogy with a football stadium. So if you watch a football match, a human being has around 1 meter in size, order of magnitude, and the football pitch has about 100 meters. So this is a size mismatch of 1 in 100. In fluorescence microscopy, you are a factor of 2 worse in just one dimension. So if you would watch a football match with a 100-meter resolution, this is what you would see. Almost nothing. If you are lucky, you could tell there's a football stadium there. But there's no way to tell what the players are doing. There's no way to tell if they are playing together, playing football or dancing or fighting each other. And luckily, a few decades ago, this was partly solved with super resolution microscopy. And this allows us really to see with much higher detail biological structures. Here you see an example of microtubules in a neuron. On the outside of the circle, it's confocal microscopy, and inside, one super resolution technique, STED in this case. And you can see that microtubule bundles that were not possible to visualize before, now you can discern them. You can really see them. Here you see another very nice example from the group of Stefan Hell. Here, 2 proteins of the nuclear pore complex were labeled, and that's how they look in confocal microscopy. Now, if you apply super resolution, this image transforms into this. So the difference is really impressive. That's before and after. So super resolution microscopy is a true discovery tool. There's no doubt about it anymore. And another wonderful example is the membrane-associated periodic skeleton of neurons that was discovered in the lab of Xiaowei Chuang. And since then, it was confirmed in all types of neurons investigated so far. So you see on the left the original image, and on the right one image from our lab. So you see, if you would image this protein with a conventional microscope, it looks like the protein is perfectly distributed uniformly all over the cell. But when you apply super resolution, you discover that that's not the case. The protein is organized very regularly with a period below the diffraction limit, and that's the reason why it looks so uniform. So this is really a discovery, and it actually is such an impactful discovery because this structure has been found in all types of neurons, in all organisms studied so far. So basically the whole neurobiology needs to be revisited in view of this, because everything we have thought so far about how neurons work, we're not taking into account this structure that it's everywhere. So you need to rethink everything. These kind of discoveries are being made nowadays with super resolution microscopy in different fields. And that's why the Nobel Committee awarded 3 of their pioneers with the Nobel Prize in Chemistry in 2014. Okay, so super resolution microscopy is really wonderful, and you have a wide library of protocols that you apply in scanning microscopes, like confocal to photon microscopes, and in wide field microscopes, and they all work very well. Before I continue, just a word of caution, because there are around there algorithms that promise you things like one-click super resolution, and you have to be very careful. That's not possible. You cannot create information, or you will create artifacts. And here you see some examples. Here you see a confocal image. That's a STED image where you reveal the periodic structure of spectrin. But of course, from this uniform intensity, you cannot reveal this just by one click. You would get very strange things. So just be careful. And now, to continue, and let's go to the focus of this talk. I promise we will try to bring super resolution down to the molecular level. So I showed you, that super resolution really reveals subdiffraction features, but it has a limit. And that limit is not fundamental, but it's practical. So if you see this beautiful image of the MPS, you will find that the resolution there is laterally of a few tens of nanometers, and in the axial direction is a bit worse, 2-3 times worse. If you look at these microtubules, of course you can see them much better with super resolution, but if we would like to zoom in and look at the supramolecular structure of microtubules, the image gets blurred again due to this resolution limit. It is known from in vitro experiments and X-ray crystallography and that microtubules are formed of this quasi-helicoidal structure of alpha and beta tubulin. But if we want to see that structure in cells, we cannot. So the resolution is not high enough. That has motivated several groups around the world to try to push super resolution to reach a limit beyond 10 nanometers. And that's not because it's a round number. That's because 10 nanometers is about the size of structural proteins. So if we can surpass that limit, then we will be able to start seeing how proteins assemble in supramolecular structures to form these higher hierarchy structures that give rise to subcellular structures, cells, tissue, organs, and so on. And the limit to that actually is not fundamental, it's just practical, it's technological. The real limit is that fluorophores, fluorescent molecules, cannot give you infinite photons. So molecules, when you try to extract fluorescent photons from them, you excite them, they return to the ground state and give you a fluorescence photon, and they do that millions of times. But eventually, they use that extra energy to undergo irreversible photochemistry, and the experiment is over. So you have a limited number of photons you can get from a single molecule. And that's the real practical limit. So there's basically two ways to solve this. You either get more photons, or you either get more information from the few photons that you have. And both avenues have been explored and have been successful, actually, in achieving higher resolutions. In getting more photons, you could think of that as the chemist approach, right? And one very successful approach has been DNA paint. I don't know if we'll have time to discuss that in detail, but there have been advances also in stabilizing buffers, self-healing dyes, and other chemical approaches. The second avenue is getting more information from the few photons that you have. That's probably the most physicist or engineering approach. That consists of developing new methods that can give you, with the few photons you have, a better resolution. And that's where our group has worked over the last 10 years approximately, and we have been successful in a few methods that can give you a resolution below 10 nanometers. I will now show you very briefly some of those methods and illustrate what you can achieve with them. So basically I will discuss three approaches very quickly. The first one is called simpler. And so this acronym was made on purpose, of course, because it's a really simple method which consists on calibrating a TIRF microscope. Just doing that, you can achieve a very high resolution. A TIRF microscope is a fluorescence microscope that uses total internal reflection to excite the molecules. So the light doesn't propagate into the sample, but you illuminate it with an evanescent field that is maximum at the interface of your sample and decays exponentially towards the interior of the sample. It looks something like that, the intensity of the light. So that would already give you an effect that would make molecules brighter when they are closer to the interface. But there's something else. Molecules also emit differently when they are closer to an interface. A molecule that is closer to a dielectric interface emits more photons into the higher refractive index medium, in our case the glass substrate and our objective. So we have a second phenomenon that makes molecules brighter when they are closer to the interface. So if you combine the two properly, you can calibrate your microscope. I will not get into details, but trust me, it's quite simple. And you need just to measure the intensity of a molecule exactly at the interface, which is easy. You just throw antibodies or fluorophores and you measure the average intensity of them. And now you can extract the axial position of a molecule just measuring intensity. And it works very well. Here you see an example of microtubules observed by Storm, and this is a 2D image. Now applying this method you can convert it into a 3D, and you can clearly see the cross-section of microtubules. So here you are seeing the lateral cross-section of microtubules, which have a size of about 25 nanometers. When you label them, they are a bit larger, but these are very small structures that you can resolve on an optical microscope. You can also see microtubule bundles that otherwise would look like a single microtubule. And now, when we looked at this, then we had a resolution of about 8 nanometers. So we thought, okay, we can see the quasi-helical structure of microtubules. Let's take a look. So we were very careful to look for straight microtubules in the XY plane, in the XZ plane, and try to look at the microtubule internal structure, but we couldn't see it. We couldn't see it. And that's because the labeling efficiency, the labeling density is not high enough. But nevertheless, since we have the resolution, we can apply very simple statistical methods like quantifying the distance to the first 5 neighbors, and then we see that those distances peak periodically at about 8 nanometers. So we can reveal that there is an underlying structure with a periodicity of 8 nanometers in this case, confirming what people have seen in vitro and electron microscopy and X-ray crystallography. So you see, this is one first example, and I will give you another one later, that reaching this level of resolution allows fluorescence microscopy to enter the realm of structural biology. And that's something really interesting that I think will develop more and more in following years, because we can perform experiments inside cells at room temperature in liquid environment, and this allows a new dialogue between structural measurements and simulations. Well, in this case, this method is just calibration and software, so if you are interested in applying it, you can download the software and also contact us if you need help, of course. All right, another way of achieving this kind of resolution or measuring molecular interactions with fluorescence is a combination of STED and FRET. So probably all of you know FRET, so I will not go into details. But I will show you this example. I will use again this wonderful example of the MPS of neurons. So the working model for this is actually, it looks like that. We think this structure is made of actin rings that are separated very regularly by spectrin tetramers. That gives this spacing of 190 nanometers. And we believe that adenosine, another protein, is actually performing the link between spectrin and actin in those rings in this way. So this is the working model. So how can we test it? We would like to know, for example, an important piece of information would be to know if actin and adenosine are actually interacting in these rings. So that very simple question could be resolved with FRET, you would say. Yes and no, because the problem is that if you would perform a FRET experiment in a conventional microscope, then you would be measuring something like that. So even if you detect FRET, there's no way you could tell if adenosine and actin are interacting inside the MPS or outside, because you're averaging a larger area. So our idea is to combine FRET, but to measure it inside a subdiffraction area limited by STED. And this already gives you a much better information, because now you can tell inside the small circle if molecules are interacting. So I don't have time to give you all the details how this works, but basically it's a two-color STED measurement. We tested this using DNA origami. It's a wonderful technology to test super resolution methods, and we could confirm that we could see and super resolve interactions through STED. And you need to perform some controls, but here you see a result. Here's a STED measurement, a FRET measurement, diffraction limited, and you would see that adenosine and actin are interacting somewhere in the cell. But it's very hard to tell anything from this. But when you combine it with STED, then you can find that the interactions present the same periodicity as the MPS. And then you can confirm that that's an active site for interaction between these two proteins. All right, and now the last minutes I have, I will use them to show you a family of techniques that we called single molecule localization with sequential structure illumination. And this all started trying to improve the localization precision of a single molecule using fewer photons. So the typical way to localize a molecule is using a camera. So you illuminate your sample with a uniform intensity, and then you use a structure detector, like a CCD camera, to collect photons of a single molecule, make an image, and then you would pinpoint its position by fitting some function, right? And this works, of course, but the localization error follows this formula. It basically, of course, it improves with the square root of the number of photons you collect, but here in the numerator, you have the PSF, you have the resolution limit. This is about 200, 300 nanometers. So you're fighting these 300 nanometers with photons. That's a problem I described at the beginning. So we tried to find another way of doing this and getting rid of this large number in the numerator. And one way we found was localizing the molecules using a light minima. The first implementation of this was a min-flux, and the idea is to kind of triangulate the position of a molecule using a minimum of light. Since we used this beam with a circular symmetry, we needed to use 4 positions, not 3, to localize a molecule in 2D. And this works very well. Here you see an example. Here below, you see many localizations of a single molecule using just 50 photons. And you see the cloud of localizations dispersed over 10 nanometers only. If you use 500 photons, then your localization error becomes already 3 nanometers. And if you can use a few thousand photons, you already get into the 1 nanometer regime and below. This works very nicely. But the key point here is that, of course, the error still scales with the number of photons, just like before. But now if you look in the numerator, we no longer have the diffraction limit. We have L, which is an experimental parameter. L is actually the distance between the minima. And we can make that smaller and smaller if we need higher localization precisions. When this was first published, it generated a big impact, especially because of this possibility of zooming in. Here you see the localization error as a function of the number of photons. They all scale with the square root of n, of course. But what you see here, these 3 curves, are just localizations made with a decreasing size of the field of view, L. So with the same number of photons, we could get something like 5 nanometers using an L of 150 nanometers. But if that's not enough, we can zoom in, and then our localization error becomes 3 nanometers. And we could zoom in farther to 50 nanometers, and then we get into 1 nanometer. So it's a very powerful method to localize more and more precisely at the expense of measuring in a smaller area. And this can be applied for imaging, like you see here for this, where we tested it with a DNA origami. You see the scale bar here is 10 nanometers. So we can clearly resolve things below 10 nanometers using an optical microscope. But it's also very useful for tracking, because since you have to localize things with fewer photons, now you can track faster. In this example that you see here, we were tracking ribosomal proteins with a modest spatial resolution of 50 nanometers. But to achieve that, we only needed 9 photons. So we could track very fast. And actually, for tracking, it's better to have higher temporal resolution than spatial resolution. That's why we chose that combination. And that allowed us to get a diffusion coefficient at video rate. So this is D as a function of time with a 30 millisecond resolution. So instead of having 1 image every few tenths of milliseconds, we had tens of images. And we could see how proteins diffuse, stop, diffuse again, and we know, of course, where it did it. So in terms of instrumentation, min-flux, it's quite complex. The first implementation was using electro-optic deflectors and electro-optic modulators. The setup looks simple, but trust me, it's quite complicated, and could achieve this measurement at 8 kilohertz. So that motivated us to make a simpler or at least easier implementation using pulsed lasers, and we did that a few years back. And that allowed us to measure this at megahertz. So now we can make these measurements like a machine gun, and then eventually we detect the photon, and we can know, using time-correlated single photon counting, we can know from which donut came that photon and reconstruct the position, and at the same time we get the lifetime of that molecule. And that opens up also new interesting possibilities. Here you see some wonderful examples that we did with a group of Professor Tinefeld in Munich, where two molecules, spectrally identical, could be localized simultaneously, just multiplexing with lifetime. So I think this is the first time where two molecules could be super resolved without blinking. And that gave rise to several different possible applications that I don't have time to go through right now. We could even, using the lifetime and position information, getting to the Fred regime of these two molecules and still find their position below 10 nanometers. So, I think I have just a few minutes. So, I want to stress that the key point of these methods is basically on using a minima of light. So, this idea of zooming in further and further to localize a molecule more and more precisely it comes just from using a minima. So what you see here is a parabolic minimum, right? If you zoom in, so if we expand this area and we rescale it, that minima looks the same. And if we do it again, that minima looks the same. And we can continue doing this forever. It's a scale invariant problem. So that's the key. And then I showed you that the first time we used this triangulation thing to localize a molecule inside that minima, but actually, since the key is the minima, it doesn't really matter how we probe that minimum. So we can probe it with these 4 points, but we can also probe it with an orbit, or we can probe it with a much more common raster scanning, like in any confocal microscope. And that works just as well. A few years back, we made a theoretical paper where we evaluated all kinds of methods based on this concept using a light maxima and a light minima. And all methods using a light minima allow you to zoom in indefinitely and achieving these types of localization precisions. So from all the methods that we evaluated, of course this one, RASTMIN, came to our attention because it can be implemented on any scanning microscope, which are widely available around the world, like confocal microscopes, confocal or two-photon microscopes. So all you need is a scanning microscope. You need to add some stabilization, and you need to modulate the phase of your excitation beam in order to have a focus with a central minimum, not a central maximum. That's all you need. And once you do that, all you have to do is to scan a small area. So here what you see are single molecules on a scanning microscope where you changed your focus to have a central minimum instead of a maximum. Then all molecules look like donuts, of course. Then you pick one and you make a small scan of about 100 nanometers. And you make that scan, and of course the molecules looks like that. It looks like a minimum. And you can localize it using 200 photons or 900 photons or a few thousand photons. The minimum, of course, looks better and better. You would say this is almost the same as localizing with the maximum, but it is not, because the key, which is the difference here, you can zoom in more and more, and the minimum always looks the same. So the error will scale with the size of your scan for any given number of photons. So here you see and some experimental data to confirm this. Here we localized molecules many times using different number of photons. The red curve is the theoretical limit, and the gray shade is actual practical limit, which considers the theoretical limit and how good we can stabilize our microscope, which in our case has an error of about 1 nanometer also. And then you see the experimental points, which fit quite well. So let's see if we can use this for imaging. All you have to do, as in any other case, when you have a localization technique, you need to let molecules blink now and localize them one by one. And again, we use DNA origami to test this. In this case, an origami with 6 fluorophores. So the origami, when they look like that, again, like donuts, then we pick one and we scan continuously a small area. And we let molecules blink, and you see the experiment looks like this. So you're scanning a small area, and eventually a molecule switches on, and every time it does, you can localize its position by fitting a minimum. And if you do that many times, then you end up with something like this. You clearly reveal the position of the fluorophores. And this is just to tell you this is done in a scanning microscope just with very few modifications. Okay, they are telling me that I have to hurry up, so I will speed up, here you just to compare the levels of resolution that you can achieve. And diffraction limited image first, then super resolution of the first generation, as we call them, and then RASTMIN or MinFlux or one of these super highly precise methods. And you can really reach about 1 nanometer and below resolutions. So to finish, I want to stress this new direction that I think it will become more and more interesting the next years, which is addressing structural biology using fluorescence. And here you see 3 papers from other groups, particularly for the group of Stefan Hell and Ralph Jungmann, where this is starting to happen because the resolution is high enough to visualize structural details of biomolecules at room temperature, in liquid environment, and sometimes even in live cells. And to finish, just an example from our lab where we addressed a new dimension of this. So positions are very precise, I showed you. Another interesting dimension of these measurements are orientations. It's very important to determine orientation of molecules. And here we show one way of doing it where we detected the orientation of a single fluorophore inside a protein cage. And this is a very small protein. It has about 8 nanometers and 6 nanometers has the size of a typical structural protein, and we could put the fluorophore inside and detect its orientation inside that cage. Which to our, I cannot explain the measurement techniques, but it's not so complicated. What I really want to stress is that we could determine the position of that molecule inside that protein cage, which is quite small. This was done at room temperature and in liquid, so we could see actual dynamic behavior. We see the molecule jump between orientations, and we found out that that molecule takes only 2 preferential orientations, and we could detect them. And when we compared that to molecular dynamics, we found a quite good agreement that is really surprising and encouraging, because now we have measurements that can be compared in a much better, in a much fair terrain with molecular dynamic simulations, because typically we have X-ray crystallography or cryo-EM data obtained in superstatic conditions, frozen conditions literally, and we compare them to molecular dynamic simulations, which is the tool we have to explore conformations. Now we have a tool that works at room temperature, liquid environment, so we can compare this to simulations much more fairly. So I think a new dialogue between experiments and simulations will be established, which of course it requires that each experiment needs to be designed to probe a particular feature of that structure. It doesn't have such a throughput as X-ray crystallography, but for particular details, I think it will become a very interesting and complementary technique. All right, with that I finish, and another advertisement of our workshop that was last week, and I hope we can hold it next year, so stay tuned for those of you who are interested in super resolution microscopy. And with that, I thank my group, and I'm happy to take any questions you might have.
Thank you, Fernando, for a wonderful presentation. So I'm glad to see that you're moving very fast to the structural biology field. And as you mentioned, I think it's a very powerful combination of techniques, especially if you combine with things that, for instance, Jose Maria Carazo does with deformations and mathematical models to predict protein dynamics. And one thing that I was very impressed that you can localize very well the protein in the Z orientation, right, Z direction. And this is a big problem when we do correlative work with cryo-EM. So how do you see you applying this type of technology to freeze down, so to localize a molecule, then freeze down the cells and go to the cryo-EM and localize them precisely?
Oh, you mean on the same sample to make
exactly.
like a correlative microscopy?
Yeah, correlative.
Well, I don't know, honestly. In my view, these methods will complement cryo-EM data. So cryo-EM and X-ray crystallography still give you a very high throughput and higher precision, right, down to a few angstroms. I think this method will be helpful to confirm structures. And like if, okay, you have some structure that you found in X-ray or cryo-EM, say, okay, I want to see if that structure really is present in a cell under physiological conditions, then you could design an experiment to probe key features of that structure and confirm its existence. I think that's the main application of this, for the moment at least.
Yeah, I agree that it's really powerful, this complementation, and on the side of cryo-EM, there's the advantage that you don't need to design a probe, right?
Exactly.
The structure is there.
Exactly. The structure is there, and so the throughput is higher still. Here you need to design a probe and a labeling strategy, particularly to test that the existence of that structure. Yeah.
Thank you. So a very, very nice question. And talk in general, and certainly we are looking for an integrative approach that mix different techniques and give a better understanding of what is going on. In terms of cryo-EM, really something that we are missing normally is the time domain. You know that there are time cores. I mean, it's difficult and all that. And when we think in time, we are thinking to use molecular dynamics, but it has to be fully atomistic molecular dynamics, because otherwise you don't have the time information. So the systems have to be small. So we are in a catch-22 there.
Right.
So.
No, that's a very good point. So the system has to be small, and also the time scales probed are small. So when you do a molecular dynamic simulation, it runs for very short times, usually much shorter than any time resolution that we have in the experiment. That's actually, it was quite surprising for us, because that the agreement was so good. Oh, sorry, my presentation. Okay, because our measurement resolution was of a sub-second, right? But it's milliseconds. And the molecular dynamic simulations runs for a few nanoseconds, right? So there's a still in the time domain. But it seems that the conformations that we found to be stable on the few nanoseconds also in the seconds. So those are really preferential orientations. But that is something that now we can probe, you know, because now we can see, okay, what we see in MD, how real is that? But it's just starting, so this is the first time we do it. Now we would like to change. This was I didn't give you the details, but this was a fluorophore with 2 sulfonate groups that we expected to form hydrogen bonds inside the cavity. So something we could clearly do now is to change those functional groups, or leave only one sulfonate group, for example, and see if one of the orientations is broken or weakened. So I think now we need to start collecting data and building a whole formalism and way of reasoning in these experiments.
Absolutely. Thank you very much.
Thank you, Fernando. We move to our next speaker. It is a great pleasure to introduce Professor Dr. Cliff Brown, a leader in the field of quantitative bioimaging. Dr. Brown's pioneering research focuses on biophysical methods to explore the complexities of cell adhesion and migration, and she has made significant contributions to advancing live cell imaging and establishing global quality control standards for light microscopy. As the director of the Advanced Bioimaging Facility at McGill University, Dr. Brown oversees Canada's top imaging center, supporting over 120 laboratories and pioneering training programs. Her influence extends beyond her institution. She is the chair and Canadian representative for Global Bioimaging, a founding member of Canada Bioimaging and the Canadian Network of Scientific Platforms, and she co-chairs Bioimaging North America. Through these roles, Dr. Brown has played a key part in fostering a collaborative international community, and she is a dedicated advocate for technology training, education, and professional development for imaging scientists worldwide. Please join me in welcoming Dr. Claire Brown.
So thank you for that kind introduction, Leah. It's a real pleasure to be here with everyone today. And I'm going to tell you a little bit about global bioimaging and different initiatives going on with that community that should also benefit those who are in attendance here. So I'll just wait for my slides. So global bioimaging is a real international network that really covers the globe, and it represents 14 partner networks in 65 countries. So it really is international. You see Latin America Bioimaging is a key member of the network, as well as the Argentinian Microscopy Network. And I'm here also representing, as Leah said, Bioimaging North America and Canada Bioimaging. So the network is organized around working groups, and these groups are really set up to address challenges within the community as a community. So really bringing people together around different topics and things that need to be discussed and sorted out, and using the collective knowledge of the community to progress. So I'm going to mention just a few initiatives in some of these working groups. So one of the key things the community has been doing, which I think is really important, is they've been publishing international recommendations on different topics. So the first one here is on image data, and how can we put together fair image data that can be shared amongst the community internationally. And this was published a couple of years ago and is available on the GBI website. So you'll notice I have QR codes on all the slides. If you're interested in these documents, you can find them. The Career Paths Working Group spent a lot of time from 2019 until about last year putting together an international recommendation for career paths. And I really love this figure, which is part of the paper. Both of these figures really show the people who work in imaging facilities, whether they be light microscopy, electron microscopy, and in Canada we also have a group looking at infrastructure facilities that cover all technology areas. These people are really integral to the fabric of science, and without these people, the advanced technologies are really, large paperweights. So we really need to recognize these people, all the different things they do. They're holding many, many hats: science, administration, management, training. And the goal of this document was to really put it down in paper of the kind of things that they do, and then also present some case studies from around the world where there are clear, stable career paths for these people who are really contributing to scientific discovery and innovation. There's also an international poster that you can put up with a recommendation on authorship and acknowledgement, so that when people contribute to research projects, the facility is acknowledged, or if they've given a real intellectual contribution to the project, that they are co-authors on the paper. And Global Bioimaging partnered up with the Royal Microscopy Society recently. The Royal Microscopy Society had developed this recommendation a long time ago, but now they've expanded it into all these different languages, and it's also available in Spanish. So there's also an international recommendation on measuring impact of facilities, and we're going to have a workshop on this afternoon, so I won't spend too much time on it. But it really is telling people how they can measure the impact of their facility, and it gives you guidelines of sort of easier things to measure, things that may take a bit more investment and time to measure. This document kind of got hidden, I find, on the Global Bioimaging website, but it's a really valuable one as well. This came out of an annual meeting during the pandemic, where we really thought we need to show the value of facilities. And it's really well written, it's very short, and it gives bullet points of the importance of facilities for different stakeholders. So for instance, for the companies, many who are here as sponsors, if the instrument is in a well-managed facility with experts, they know that the instrument's going to be performing well and bringing out good data for the customers. So it goes through all the different types of stakeholders and what the value is to help you make a case, I guess, if you're talking to your administration or your funders of the importance of these facilities. I wanted to also mention the Training and Education Working Group. I know that they have a good connection with the Training and Education Working Group in LABI, and Marcela Diaz is one of the co-chairs of that group, as well as Mariana Dinez, who's I know is an active member of LABI as well. And they're working on generating training resources, collaborating internationally, making sure materials are translated, and so on. There's also quite a few training resources through the Global Bioimaging community. So they have a lot of training activities for facility staff. They also have an international job shadow program, and I have the QR code there. Anybody working in an imaging facility anywhere in the globe can apply to get funding to travel to another facility for professional development. If you want to go and learn about facility management, or you're getting a new microscope and that another lab has the system and you want to learn more from them, and so on. So it's a really valuable program. And Leah actually just took advantage of that program to come and visit my facility after the Global Bioimaging in Montreal. And then there's a really great online training platform for independent learning. So you can go, there's dozens of modules where you can go and learn on your own time. They're guided through on all different topics of microscopy, facility management, and impact. So this is just an example of some of the training courses. They've really been run all over the world, all different topics: image analysis, facility management, train the trainer. And it's really a great community if you can get involved, if you're not already. And I wanted to also mention Imaging for All. This is a program that was recently funded by Wellcome and is run by Global Bioimaging. And they're going to have another call in December. And there's 3 different levels. You know, you can travel to go to a course or a conference, a workshop. You can also travel to do experiments, so go for 2 weeks to a lab and bring your samples or prepare your samples there and collect data. And there's even a pro one where you can go quite a bit longer. And one thing I would like to emphasize here is I think a lot of the programs have been traveling to North America or Europe and so on, but you can use this funding to travel within the region. So we've just seen from our keynote, you know, there's amazing technology and expertise here in Argentina, in Brazil, in Uruguay. So you can apply for that funding to travel within the region as well, and you know, gain the local expertise, and you can fund your travel, the fees for the facility where you're going, and so on. So it's a really great opportunity. And I wanted to mention the microscopy database. This is an international project that we've taken advantage of in my facility as well, and you can post jobs, events. If you have CAD drawings of cool live cell chambers for your stage, or expansion microscopy, adapted things to hold your large samples or whatever, you can upload those under the tools. If you have software that you're trying to promote for people to use for image analysis. And the idea is you put it in the database once, and then we've partnered with all of these organizations across the globe. And so if you put your job ad there this afternoon, it's going to be in Canada and Singapore and Australia tomorrow on their websites. So it's a really great initiative, and I know for us we've been getting a lot more international applications for our courses since we started advertising through this. And then lastly, I just wanted to mention technology user groups. So this is not a global bioimaging initiative, but through Canada Bioimaging and Bioimaging North America. And we've been trying to get people together around advanced technologies to discuss best practices, but also talk about things that don't work. That often doesn't get into the paper, and people waste time repeating something that someone else knows very well is not going to work. So we have, regular meetings on Zoom, we have guest speakers, we have brainstorming sessions, and right now we have 4 topics. We have STED microscopy, expansion microscopy, lattice light sheet, and we also have a metadata and data management group. So if you're interested in getting involved with any of those groups, we try to keep it light, we try to keep it really targeted on the needs of the community. And also if you're working in any of these areas and you want to present your work or present a project and get some feedback, are you on the right track? It's a really great community as well. So the main event of Global Bioimaging is the Exchange of Experience. And this is run every year, and we just had our last meeting in Montreal just a month ago. And the next one is in Heidelberg in October 2026. So I just thought I would mention that in case people are interested to join the community there. And I think that's my last slide. So I'm happy to take any questions. And if there are no questions, I'm around for the duration of the meeting, so we can talk over coffee.
Claire. Okay, I'm just very interested with BINA at the moment. So can you say something about funding, since things are sort of disappearing, and how you're addressing that?
So Bioimaging North America has one more year of funding, and then we'll be looking to try to renew, but it's pretty unclear, I think, whether the Chan Zuckerberg Initiative is going to continue to fund the networks. So I think, obviously, a lot of the tools we have here are to advocate that these networks and these facilities are really important, and we're working on that. But I think in the global climate, it's a little difficult to pivot. I think brainstorming, looking at other options. I know the Imaging for All has been quite successful, so perhaps Wellcome will continue those initiatives. And recently we applied for a grant from the Spencer Foundation, which I had never heard of, but their focus is on education. So we applied for some of our training programs, and I'm happy to provide that. But I think that's another, one of the other working groups with Global Bioimaging is how to talk to your funder. And I think that's something else we could do, is also get like a database of potential funding, philanthropists or different organizations. The Spencer Foundation is a nonprofit. So yeah, we're working on it. Happy to chat more, but it's definitely challenging times.
Just a follow-up question, Claire. First of all, thank you so much for your presentation. So you've been in Uruguay and for a while in Argentina, also in Brazil, very briefly. So you know very well our community. So a follow-up question, how do you see that LABI could position itself more strategically to face these challenges that we have worldwide?
Yeah. Well, I think just my comment about Imaging for All, I mean, it could be very simple things, like don't apply to go to Europe if you don't have to. If you have the technology and expertise here, your travel costs are going to be less. You can spend more time in the lab with the funding to do the experiments. So I think that was something that was very useful for me to learn, of the level of technology and expertise here, which we just don't hear about in Canada. So I think don't be shy to talk about all of the excellence that you have. I think I definitely learned there's excellent scientists and people are very, very well trained. I mean, the universities here give a very strong training. And I think students should be sought after and so on. So I think being creative with the funding that is out there. And Lee and I were talking about whether we could set up, BINA has quite a bit of funding for exchanges, but they're usually within North America. But maybe we could talk about earmarking some of that funding for exchanges between North America and Latin America, whether either direction, And then I think looking at different funding options, like we're looking at making some partnerships between University of Buenos Aires and McGill University, for instance, to see if we can get funding from some of the Canadian international initiatives and so on. So I think we have to just keep plugging away and keep looking. I think there's a lot of funding out there that we don't necessarily know about. And I think the tools Global Bioimaging has adapted to show our impact and measure it and have data to say it's important to fund these initiatives because they're having an impact on science. Okay, thank you.
Okay, thank you very much, Claire and Fernando, for your presentations. Now we move to the next session, the roundtable. So please, I call to come to this space Kildare Miranda as the moderator, and Carolina Perez, Sergio Denner, Juliana Oliveira, y Francesco Rossi.
So good morning, everyone. So now we'll start our first roundtable, which is entitled Democratizing Advanced Technology in Latin America. And so this session is dedicated to a dialogue on the barriers, strategies, and sustainability models to advanced imaging access across the region. We will discuss topics that include infrastructure models, open hardware, sustainability, policy advocacy, and innovative funding mechanisms. The participants will explore strategies for resource sharing and technology adoption. Open exchange period allows the audience to participate and have a regional perspective. So please participate and ask questions. So we have here today, it's a pleasure to have Carolina Perez from the Dirección Nacional de Infraestructuras de Investigações Científico-Tecnológicas da Secretaria de Inovação, Ciência e Tecnologia. Juliana, vou falar em português, do LNBio, do CNPq, do Centro Nacional de Pesquisa de Energias e Materiais no Brasil. Sérgio Derra, da Bioengenharia, e Francesco Matias Rosas do FALAN, da Faculdade de Ciências Udelar no Uruguai. Entonces, let's start with Carolina has a presentation. So, please, Carolina.
Hello, good morning for everyone. I'm very glad to be here. I thanks.
Is it working?
Okay. I thanks Leah and all the organizer committee for this invitation. So I'm going to start to run with my presentation. Give me a shot. Well, the topic of this roundtable, I want to say something about the national system, which is an initiative created in 2008 with the creation of microscopy national system, and their principal objectives are to coordinate efforts and promote the efficient use of physical resource, optimize the operation, and facilitate the opening of their service to the scientific community and the productive sectors. With more than 10 years of operations, I want to highlight some of the more important results of this initiative, which are this national system made the use of large scientific equipment more efficient and productive and accessible. They promote a new form of collaborative work between institutions, and they mark the beginning of a cultural shift in the share of use equipment. The most important tool of these results is the Shift Managed System, as we call Sistema Nacional de Turnos, or SGT, which is a system for managing the activity of equipment and allows measured time of use and the availability of the equipment. From the roles of the administration, they can configure the laboratories, the schedule time, and condition of use. From the roles of the user, which is open for everyone who wants to use this type of equipment, they can switch the equipment which suits for their needs from a map or a forming list, and they ask an appointment very easily. So now we are working in a new approach of the national system, opening to the all of kind of infrastructures, such as scientific and technological research infrastructure, which include larger scale and strategic equipment, scientific and technological facilities, and larger scale of scientific research instruments. This is why, because in the last 4 years, the Ministry of Science, at that time, now we are a secretariat, have improved a program which called Equipar Ciencia Program, who purchase a large type of equipment. We need to incorporate almost 500 equipment from different types. So it's important to change the approach of the program to make it to achieve to all the type of equipment. Now talking about specifically of national microscopy system, I want to show you some of the effort of almost 20 years. We have incorporated to the system almost 200 equipment from different types, as you can see, and 252 microscope that has to be incorporated, which I purchased it through the Equipar Ciencia program. Now I show you these graphics that shows the different type of equipment that now are in the system, and you can use it through the SGT. And these are the types of different microscopes that are buying through Equipar Ciencia program. And the last slides, I want to show in the last 10 years the use of the equipment through SGT, like requests, users, and hours. I think that this tool is very important for all the system and for all the type of equipment, because it is accessible for everyone. Of course, we have to work very hard in improvement of the systems. It's very old for the new technology, and open it to all the type of research infrastructures. And I think that the collaborative way to work is the most important to achieve that objectives. Thank you very much.
Thank you, Carolina. So now I'd like to ask our panelists to give a brief 30 seconds introduction of what you do or your institution and what the projects that are currently involved. So, Juliana.
So, I am Juliana Oliveira. I am leader of division in LABI Bio. It's Brazilian Bioscience National Lab. And it's at CENPEN, the Brazilian, the Center for Research in Energy and Materials, where we have the CIRUS.
Sergio.
Buen día a todos. Bueno, mi nombre es Sergio Denner. Trabajo en bioingeniería en el área de servicio técnico y representamos a SAIS acá en Argentina.
Okay. Francesco.
Hola. Hi, my name is Francesco Rossi. I'm here as chair of the executive committee of FALAN, that is the Federation of Neuroscience Societies of Latin America and the Caribbean. And I'm also a neuroscientist at the Faculty of Sciences in Montevideo, Uruguay.
Okay, thank you. So then we'll start a very brief or quick round of questions to each one of the panelists. So we'll start with Juliana. Could you talk a little bit more about the projects that you have in LABI Bio, specifically the Orion project, right? Which is a world-class investment in bioimaging, integrating synchrotron light with biosafety level for laboratories. And so from your experience, how do you balance the ambition of being a global reference with the responsibility of democratizing the technology for Brazilians and Latin American researchers and that do not have access to this type of technology? How do you see that?
So, thank you. It's a pleasure to be here talking about LABI Bio and CENPEN. And the Sandpans mission is exactly to provide scientific advanced technologies. With our facilities are open. We don't have charts for academic users. So we believe that this is very important for democratizing science. So in our mode of operation in LABI Bio also, and Orion will be the same way. And we are doing other things to democratize science and also for inclusion to reach other regions, mainly in Latin America. So we have financial support for users abroad, São Paulo State. And some examples, we have more than 10 different proposals from Argentina at LABI Bio, only at LABI Bio, so we are very happy. And we believe that these financial support are very important. And also we are doing calls directed to areas from north of Brazil. For example, we launched a call last year to provide biomolecules, oligonucleotides, for these regions. That is a very important material to do science, a good science, right? And label materials, label oligos, fluorescent oligos. So our proposal is exactly this, to democratize the science, and we believe that the partnership with LABI is very important also for Latin America.
Yeah, just a bit of context. So LABI Bio is they already built a generation 4 synchrotron, and now the Orion project will integrate a biosafety level 4 laboratory with the synchrotron. So we'll be able to perform experiments that we were never able to do it before. Yeah. But this leads also to, so it's a terrific model for the different countries, but also requires a massive investment, right? Yeah. So how do you think that LABI Bio and the model that you are developing could serve not only as a cutting edge infrastructure, but also to as a catalyst to elevate the research within the region?
Yeah, we believe that we're doing trainings, receiving people, users, and also participating in these partnerships like LABI and GBI. So Tainá, that is our scientist from Bioimaging Lab at LABI Bio, that is here with us. She went to the training the trainer, and she is now training the users of LABI Bio. And we believe that in this way we can propagate and knowledge.
Okay, thank you. So I have a question to also to Sergio. So from the industry perspective, so Zeiss and other companies, we know that they recognize that Latin America is an important partner. But the companies also know that we have unique problems, and sometimes sustainability comes as a main barrier here. So beyond the traditional equipment sales model that we already are familiar with, what strategies you think that the industry may apply to explore and make advanced technologies accessible to our region?
Bueno, desde Zeiss consideramos sí que Latinoamérica es un mercado muy importante que está creciendo y se está consolidando, sobre todo con esta red de LABI. Estamos al tanto de las dificultades económicas que eventualmente atraviesan por cada uno de los países, y para lo cual Zeiss, bueno, eventualmente va evaluando las oportunidades y tratando de adaptar los presupuestos o las condiciones para cada equipo en particular.
Okay. So in the follow-up questions, so there's some tension between institutions that want to have access to cutting-edge instruments, right? And the realities of maintenance, consumables, training, technical support, etc. So how the industry can work with networks like LABI to provide support to those areas?
El tema de mantenimiento o la postventa es un punto muy importante, sobre todo en la gama de equipos que componen la red del LABI, que son equipos de alta gama. Por suerte, bueno, la tecnología ha ido mejorando y ya hay muchos equipos que la cuestión de mantenimiento de partes se ha reducido significativamente. Y por otro lado, SAIS también colabora entrenando a técnicos de cada país para tener que, en lo posible, un técnico de forma local y evitar gastos, digamos, de tener que traer a alguien del exterior. Así también hay soporte postventa monitoreado directamente desde Alemania, que no tiene costo para los equipos. No todos aplican, pero bueno, muchos de ellos sí. Y bueno, hacemos lo posible para estar junto a los investigadores todo el tiempo, que sabemos que hay oportunidades en las cuales hay fondos que normalmente son para comprar los equipos y muchas veces no hay fondos para repararlos o para el service. En ese sentido, SAIS también acompaña a los investigadores, eventualmente dando descuentos especiales o hasta incluso repuestos sin costo. Y bueno, los mantenimientos eventualmente también son sin costo. Pero bueno, así estamos. Sí.
Okay, so moving forward, so I have a question to Francisco. So, as president of Falan, you have this continental perspective of the needs of the neuroscience within the region, and we know that neuroscience also need a lot of advanced equipment, from MRIs to photon microscopy. So, how do you see the main bottlenecks? What limits advancing neuroscience within the region, considering our infrastructure and the gaps that we have, technology gaps that we have in the region?
All right. Okay. First of all, I should make it more clear what is Falan, which is just a federation that gather society of neuroscience. So let's say we don't have our own funding, so we apply to calls, regional and international calls, to develop our activities, and as many of us. And okay, so just to comment also that we recently organized, promoted and participated in a survey here through our members on the needs and difficulties to develop neuroscience in Latin America mainly, and technology limitation was one of the main. I think we had kind of 20% agreed that technology was a strong limitation, 80% completely agreed on this. So it is clear that it's quite a strong limitation for us. So in this, I had to write something here. So there is a lot of gap. What can we do? I don't know. We may talk about this later or now. But there were other questions in this survey. That was how could this technology be incorporated? Which strategies can be used and besides funding, okay, there were different ideas, and we are doing a little bit, mainly meetings and to discuss policy advocacy and this kind of initiative, and workshops, and we are organizing next year a congress where we will have a few sessions on this, and also on technologies in general, microscopy and other activities. And yes, and this is, I mean, we also try to organize what is the actual situation of technologies in general, not only imaging technologies in Latin America, so to have kind of map of the resources and the facilities that are available in Latin America and facilitate resource sharing and so on. And this is under construction, of course. And this kind of initiatives, of course, are super important. I'm, of course, super interested in many, in discussing with many of the people participating in this meeting, so to share ideas and eventually develop something else.
And how scientific societies and networks like LABI can learn with Falan to advocate, to play a role, to defend more investment and in shared infrastructure?
Well, how? In this way, this is the answer, I suppose. And I'm learning. I'm going to learn a lot from many people here. And the federation is, you know, it's a federation of societies. So it's already an achievement, I believe, that is not always very easy to gather societies together, because when in each lab it's always the same. You work for your own research project, and then you have 5% of your time that you dedicate to build something more institutional and so on. And this is a difficulty, I suppose. And the other is again, that there are different initiatives. And in Latin America, I mean, I'm discovering, I'm realizing that there are very many initiatives, and regional level and also a global level. Here we are in imaging, in the imaging world, let's say, but there are many others. And even around the imaging world, there are a lot of different initiatives, and gather together these people is super important. And so keep working like this, and keep discussing and try to find some common strategies, some common strategies. See.
Any comment on the panelists? Questions from the audience? Yeah, Lionel.
Thank you. Well, I have a question for Carolina. Thanks to be here. Well, my question is, we all know that national systems were one of the most important projects and ideas that had the other government, but unluckily, let's say, we don't have grants. We are not having grants for the last 2 years. We don't have money to buy equipments, even replacement parts for our equipments nowadays. So my question is, what are the plans for the next 2 years regarding national systems?
Well, I know that, and it's not a very good situation. We are clear. I'm work from the initiation, in the secretary in the initiation of the national system. Now I'm very excited, and I expect that with the budget for 26, we can open the lines of de financiamiento que estaban antes. Lo digo en castellano porque me va a resultar más fácil expresarlo. Hemos hecho un pedido importante de financiamiento para el próximo año. Hoy no te puedo dar una respuesta. Como sabrán, también han habido cambios en el gabinete, que eso a nivel político todavía no sé qué impacto tendrá en la Secretaría. Sí, las propuestas se hicieron. Ahora es importante comenzar a relevar todo el equipamiento que se compró con Equipar Ciencia, saber en qué estado está. Sabemos que tampoco se compraron lo que inicialmente se había pedido, con lo cual también es importante relevar con el formulario de adhesión, que estamos trabajando, digamos, en algo nuevo que también permita vincularlo con el mapa de servicios, y en base a eso empezar a, digamos, a poder tener estas líneas de financiamiento como estaban en su momento, y quizás tener alguna que sea para algún tipo de equipamiento estratégico por algún tema específico, que son los temas prioritarios que éramos la Secretaría, que son salud, agroindustria, minería, pero un poco es en esa línea. Lamentablemente hoy, desde mi lugar, digamos, no puedo decir, no puedo dar ninguna confirmación. Sí que el pedido presupuestario efectivamente se hizo, pero bueno, va a estar atado a la aprobación del presupuesto y en cuanto asignen, pero digo, desde el lado de las autoridades políticas sí reconocen la importancia de los sistemas nacionales, la importancia de los equipos, y también es algo que, digamos, que es mi intención y que venimos planteando también la importancia de actualizar el sistema de gestión de turnos, porque eso también permite que las inversiones también sean estratégicas, porque si uno puede demostrar que el equipo está abierto y que se usa, ayuda a fundamentar esa inversión o esa posibilidad de financiamiento.
Leo.
Okay. Well, thank you very much for the participant of the table. I have like 3 reflections, maybe 1 question. So one thing is regarding something that Claire mentioned in her talk, and I think, you know, speaking about democratization and taking advantage of opportunities that we have for collaborating in Latin America, stressing the opportunities of Imaging for All for Latin American cooperation, I think is very important. Me as a representative of LABI in GBI, when I saw the first statistics about, you know, how was distributed the assignment of grants to go from Latin America to North mainly, I saw this as a failure. It's a really great opportunity. I'm not blaming anybody that take advantage to go to North when, you know, specific techniques are required. But I am truly convinced, as Claire mentioned, that in Latin America there are, you know, fantastic technologies, fantastic imaging scientists with capabilities in place to lead research at, you know, global standards, right? Without competing with anybody, right? And I also see this as a great opportunity for collaboration with other networks as Francesco represent here, right? So there are facilities. LABI is in position to show everybody where are those facilities. We have already represented over 70 centers with state-of-the-art technology here and there. We are able to make that connection, and I think the engagement with FALAM and other scientific communities are key. LABI is in the position to demonstrate that. And this moves me to the next reflection, which is how we collaborate also with the industry. You know, Sergio is here, behind me is Rupert, and I know that is Leica and other representatives here. So we are all knocking the door once in a while for a course, for an organization, for a problem with reparation. When we have regional, you know, representatives that cannot support the instrument that they sell in Latin America, this is also a matter of LABI, how LABI can, you know, put together people and complain in a gentle way, but in a strong case, to say, well, we are here, we buy instrumentation, we deserve the same, you know, treatment that anybody has in the global north. So I think, again, LABI is in the position to speak with all regional representative and international representative to make easy their life, you know, to make simple and avoid, you know, over, you know, knocking the door or, you know, overwhelming all of you asking for you know, money for organization of meeting and etc. So I forgot the question, but it was something that I wanted to say.
I couldn't agree more, Leonel. We so far really have a neurocentric model in the Imaging for All program. But I think that global bioimaging is also, so Claire may make a comment on that, but I think that global bioimaging is worried about that. It's for the success of the program, it's better that we have more opportunities everywhere, right? Otherwise, we just repeat, it's another program that adds more to the programs that already exist. I couldn't agree more. And I think that from the LABI perspective, as you mentioned, so we have the infrastructure, the connections, and we can work very well as facilitators in these connections, right? We just have to structure our framework so that we have sustainability so that we can keep our programs running. So any comments from the panelists?
No, it's just a comment, but as an example that I lived in these last few years as president of the executive committee of Falan, that is we are member of the International Brain Research Organization, and we started to use the word Falan is the hub of neuroscience in Latin America. Okay, but this is important for a funding organization or partners, so on, to have one contact, not only one contact, but one contact as the federation, which can share the information for funding or whatever to many other members, instead of having many different contacts, let's say each one of the Society for Neuroscience, that is still occurring, of course. So I believe in LABI, perhaps in the world of imaging, could or should be this hub for imaging in, I mean, I'm not really in this network. I don't know the policy, the politics here, how is organized really, but LABI or this network, the Latin American Brain Imaging Network, should present himself as this, the hub for imaging in Latin America. So to facilitate, I mean, it's just a parallelism with what is happening for neuroscience. And that's it. It was a comment.
I totally agree. LABI has been very essential for us from LABI Bio. So we arrived last year, more or less, in this community, and you receive us very, very nice, very well. And now we are increasing the team, so we are doing the trainings. Now we have here Elaine and Humberto that are researchers from Orion project. So they are doing new technologies for Orion, and they will present here. So I believe that this community is very, very important for Latin America and CNPAN.
Okay, so then I have a last question for all the panelists. So what do you consider the most critical and structural barrier to democratizing advanced technology within the region? So everything counts, like funding, we already know, but other things, business models, interactions with the companies, how we connect better laboratories, how do we exchange experiences within Latin America? So what do you think about it?
Well, I think that one problem, I'm speaking for Argentina from my point of view, I think the change of governments sometimes affects the policy, the strategical policy, and that is a barrier for democratizing science. I think that is one of the problems. Obviously, budget is a big problem, and I think that one way of solution that maybe is started to signing memorandum of understanding from all the parts that integrate the national system, and this move has to be the long term, so the changes of the government can't affect that. This is my personal opinion.
I agree with you. Yeah, totally.
I can complement. I believe that the regional vision is very important to think in our problems, because no one will solve our problems, only us. So we need to be together, like LABI is an excellent example of this, because we need to do that. And I think that we need to have a strategic plan to have the equipment and the technology that we need here, right? As a community, think what the researchers need, what we need to do in strategic areas to have a good science.
Well, okay, there are at least two ways. One is to convince the government, the regional government altogether, which is a very hard way, because it's already very hard to convince one own government to support, let's say, something, science. In Uruguay we have just now strikes and so on for reducing the investment in education and of course in research. And the other are other kind of initiative, which is everyone fight for his own, its own country, its own lab, its own world, or relatively small world, and try to democratize through these networks, you know, the access to facilities and so on, which, I mean, we can try to do both in parallel. The first one has always been quite hard. I think Argentina is a very good example, not now, but eventually. And Brazil is I don't know much, but there are islands, no, of very nice places with a lot of technologies. And okay, Uruguay is very small, but the strategy to the Mercosur, I don't know, there was the FOSEM one, the grant, and it is still working, I suppose, for some kind of project or initiatives and so on. But I think we should try to work on both strategies. Support the institution with more funding to obtain huge materials and technology, and find a grant to support people from all over the region to access these same technologies on the other way.
Okay, thank you. So I have to wrap up here, we're running out of time. I just wanted to make a very quick comment. One thing that I think that Latin America is getting stronger and stronger is in open science. So we have a lot of groups that are able to produce their own instruments, to readapt old instruments, even if it's a different instruments, not a microscope, but they use lasers that were previously used in gene sequencing, for instance. And this is something that is growing a lot in our community. A number of light sheet microscopes were already built within Latin America. We just saw the example with Fernando Stefani. So I think that this is something that we should keep in mind. In our region, there's a lot to discuss it, to exchange in this regard. Sorry, I didn't see you. Yeah, okay, so last question.
No, it's just a little comment, thank you.
Yeah.
I wanted to add something to what Claire and Lionel just said about trying to improve Latin America connection. I don't know if LABI can, in order to have more visitors inside the region, could be helpful to have like kind of financial support to the hosting institutions to support visitors, something like that. I don't know if possible to have something like that in order to cover expenses and very simple supplies to receive more students in our labs now that we have a very critical situation.
Well, within the Imaging for All program, there is, right? Within LABI, we don't have it. But this is something that it's in our radar. It all comes down to funding, right? But definitely it all with Imaging for All. It depends on the structure that you have, on their size, and how the laboratory or the center will be classified to receive people. There are different levels, as Claire just mentioned. I think it's 10K and 20K, right? Depending on the position. And that's basically it. Yeah, Lionel?
Despite that LABI doesn't have now resources, we benefit from the CCI grant, for exchanges that work for training that then kick off collaborations, right, and things like that. But despite also of Imaging for All, la UNOM BioLAC, la Academia Latinoamericana de Ciencia, and the Company of Biologists, they are all opportunities that support for exchanges. They are not huge, probably for a few $1,000s, but at least cover the expenses for traveling and some expenses for experiments.
So we have to wrap up, guys. Thank you very much. Thank you very much to our panelists. And now, Leah, coffee.
So each speaker will give a 10-minute presentation, and after the presentations at the end we will have 10 minutes for questions. Therefore, please save them for the end. Yes. Fernanda? So first we would like to invite Leonel. Okay, Leonel is director of the Unidad Avanzada de Bioimagenología at the Institut Pasteur at Montevideo, Uruguay, and his work is focused on film microscopy and hyperspectral imaging. Welcome, Leonel.
Well, thank you very much. It's my pleasure to be in another LABI meeting. And today I'm going to talk about a technology that we have been developing for the past couple of years in Uruguay, the diamond microscope. Okay. Okay, so basically this technology is aiming for deep tissue imaging and working in live samples. And basically, as Nicole said, I am the head of the Advanced Bioimaging Unit. This is a joint initiative between the Institut Pasteur and the Universidad de la República at the Clinical Hospital in Montevideo, with the main goal of fulfill these four claims that we have here: provide services, training dissemination, developing technology in terms of hardware, software, and methods, blending spectroscopy and microscopy, and basically use this technology for biological question in the lab. So speaking about technology, we have several commercial microscopes with the new 990 that we just received, but we also develop other technologies that we either adopt or we basically develop in our place. And we also have capabilities for bioimaging processing with several high performance workstation, and also computing and storage server for the technologies that produce a massive amount of data. So speaking about multiphoton microscopy and deep tissue imaging, one of the issues even for multiphoton microscopy is when you want to go very deep in the tissue, you have to pump your laser. As higher you pump your laser, you start to have autofocus fluorescence due to the high rate of light that you are applying, and this basically goes contrary to the penetration. The reason for why this happens is because, well, you have a wavefront distortion due to the travel of light through the tissue, and basically the inhomogeneity in the index of refraction will make your sample very, very clean and crispy at the beginning and in C, but when you go deep, 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 basically you start to blur your image. So the diamond microscope is a technology that aims to catch more and more photon in comparison with any two-photon epi detection microscope. But before going to the diver, I want to mention that there are several other alternatives in the business that use either this parabolic mirror or fiber optics surrounding to the cone of illumination and detection in epi detection in order to increase the number of photons you can collect. And even in the best situation, you can go 2 to 5 folds gains in number of photons in comparison to the epi detection. The other possibility is always to clarify your tissue, but for most of the application that we run in our lab, we don't want to clarify samples. We want to work with samples in the native state. So for that reason, many years ago, Enrico Graton, Sasha Dvornikov, and Viera Kerstingari developed this technology. In a regular epi detection microscope, we have an excitation, and basically the optic focalizes the light into the tissue, and the 2 photon spots emit in all directions. Within the tissue, with high scattering, you have photons that undergoes in all directions, and you have only a small angle to collect the photon that you excite. Well, the diver is a very simple concept. So basically, it's a microscope that has a detector in forward, a very large area. This is a 1-inch square detector that collects photons under scattered. And the reason for why it can collect photons in scattered regime that you cannot collect in the epi detection is because you have a much index of refraction and there is no optic in between. So basically, it's like a transparent path for photons arriving to the detector. So we can collect virtual numerical aperture than any other epi detection can do. And this is an example. If you match index of refraction between slices with water, you can see through the slices, and if you don't, basically the photons get scattered. So in overall, basically you can go up to 1,000 times more signal to noise in comparison to any 2-photon microscope. So this is the detector. Basically what you need to do is to build a detector. And the detector is basically a wheel embedded in ethylene glycol with glass filters, and this is the Hamamatsu detector. It's a PMT that we use. It's compatible with custom, but also with commercial microscope. So there is only 4 microscopes, all built by Sasha Dvornikov in US, and the 5th is the diver in Montevideo, Uruguay, which is the only one outside in the world. And basically, this technology requires you to build basically all the mechanics in order to put together a microscope that looks like this. And this is the software performing and scanning, in a piece of sample for calibration purposes. So what are the benefits of this technology? Well, one of the big benefits of this technology is the enormous or giant field of view. So you can go to samples that are centimeter long. And the reason for why is because we have a scanning optic of 2 inches. So we can really steer, the beam widely than any other commercial microscope. We can also go deep, and the reason for why we go deep is because this wide detection, and basically in a very high scattering condition, like in a phantom here, we can go up 2 millimeter, basically 600-fold time increase in sensitivity in comparison to epi detection. But we cannot even be sensitive in a transparent sample. And the reason for why is very simple. As deeper you go, the virtual numerical aperture you have is increased because you are collecting photons that are emitting closer and closer to the detector. So virtually, you have different excitation and emission numerical aperture. And this is the reason for why we can go deeper and deeper. So the applications, basically, we stress the use of this technology in combination with fluorescent lifetime imaging microscopy. And the reason for why is because we wanted to use for label-free metabolic imaging. And NADH is a metabolite that we have in all our cells, and basically it can be in 2 forms within the cell, free and bound to the enzyme that this cofactor does, a job in the cell. And basically using this measurement, we can measure the heterogeneity in lifetime as a metabolic index of the cells. And using that, for instance, we can see in slices of rat lung, basically the metabolism between a lung that undergoes to damage by bronchoalveolar lavage. So basically, we can use this for metabolic imaging in cells and also in tissue. The other very 2 strong applications are also in level 3 for second harmonic generation. Second harmonic generation is a signal that propagates in forward in structure with not centrosymmetric structures like collagen and myosin, or and is very strong in urea crystal. And basically, we can use this for, studying fibrosis in kidney or separate the different collagens in the femur that I showed before in the intensity image. But now, basically, regarding to the information of the lifetime and the second harmonic generation that different collagen generates. The other thing that is very strong is for third harmonic generation, and in that case, the signal is generated basically in changes, in dramatic changes in index of refraction, and this happens, for instance, at the surface of lipid droplets, and this is an example that we did long time in the lab, where basically we can see in a liver the effect of high-fat diet and the accumulation of very large lipid droplet in the adipocytes, but also can be used for 3D imaging of sciatic nerve, combining third harmonic generation, second harmonic generation, and autofluorescence. The last thing that I want to mention is this microscope is also compatible with the hyperspectral imaging, and basically in hyperspectral imaging, we need to collect step by step the different wavelength to recompose the spectra, and then we use this in the spectral phasor analysis, which is a Fourier transformation of the hyperspectral data. But Enrico came up with an idea like 5 years ago, in which if we collect the data in a modulated spectra, basically we can do the phasor transformation on the optics, but not in the using mathematics. And doing so, we can also acquire information from the same femur, and basically here is the different spectra from collagen regarding to lifetime, and this is information regarding to the spectra that the collagen information give you. So it's also a very nice characteristic of the diver that is not easy to achieve with regular hyperspectral imaging in confocal microscopy, because scattered photon doesn't go well with dispersive optics. The final thing is the microscope is compatible with adaptive optics, and in the Enrico's lab, they also implemented adaptive optics in order to increase even further the capabilities of deep. And to finalize in my last 30 seconds, I want to thank the team that was putting together the diver, Luis Inchausti, Micaela Lopacio, Sasha Dvornikov, who was in the lab, and also collaborators over the year. In particular, I want to mention Enrico Granton, who was my former postdoc mentor and also a big inspiration for what we do at the Advanced Bioimaging Unit in Uruguay. I want to finish wrapping up saying that we are pushing in other directions. So we receive a new heart for the diver. This is a new laser with an extended range that we wanted to use for purposes of exciting other molecules, but also for other spectroscopy like wavelength mixing, cars, and things like that. We are waiting for our Flamingo, which is a fantastic initiative between Uruguay, Brazil, and Chile. And there are many friends here participating in the consortium. We are also working on the image scanning microscopy in collaboration with Giuseppe Vicidomiti in combination with FLIM. And we have implemented phasor pi and a pariphasor for all the analysis of fluorescent lifetime hyperspectral imaging. With that, I want to thank the funders, and of course, I want to thank my team, which basically the people who does all the heavy work in the lab. Thank you.
Thank you, Lionel. Our next speaker is Rupert Ecker. Rupert is CEO of Tissue-Gnostic, which you probably saw in the hall during the coffee break. And he's also adjunct professor at Queensland University of Technology in Australia. So please welcome Rupert.
Yeah, thank you very much for the kind introduction. It's my great pleasure to be here today in Buenos Aires and talk about my passion. My passion being bringing the idea of flow cytometry, which is quantitative analysis of antibody-mediated staining on the level of individual cells, bringing this idea from cell suspensions, thank you, from cell suspensions to the tissue section, the microscopic slide. And I have been working on this passion both academically, I'm a cell biologist by training, and developed a prototype of this technology for tissue image cytometry as part of my PhD thesis in the 1990s. Founded a company, Tissue-Gnostics, and then came back to academia, and now I'm working at Queensland University of Technology in Brisbane. And when we started, I made a search by the time, it was 2003 when the company was founded, on PubMed and looked for FACS or flow cytometry. And I found thousands of papers which had used flow cytometry as a tool of research and discovery. And there was not one which used tissue image cytometry. I'm not going to talk about our products. If you're interested, then please see me and my team outside at the booth, but I would like to draw a more comprehensive overview and also reflect on the basics. Probably because we are here in a bioimaging community, many of you will know this image. So the core question is about perception. And if you look on these two frames, A and B, we will probably all agree that A looks much better, much darker than B, but in fact it's not. A and B have the same gray value. It's optical illusion that makes us believe A is darker than B. And this is something we have to be aware of, that if this was a tissue section with a cell stained for whatever marker you're interested in, we would see here a cell in a high background environment and conclude its marker negative, but here we would see a cell in a low background environment and conclude its marker positive, and we would be wrong. So the take-home message of this slide is that our perception might not be determined by the structure of interest we are looking at, but by its vicinity, the surrounding. It also comes true when it's counting of cells. We see here classical immunohistochemistry staining with a well-known marker, Ki67, proliferation marker in brown, peroxidase. And the obvious question is how many cells are proliferating, or how many of the blue nuclei are also brown. We made a survey among more than 300 scientists, and believe it or not, the answers that we received ranged from 0.5 to 60%. And these were all scientists. I'm not talking about people on the street. And I think this is a significant difference, and it shows the main problem associated with visual evaluation. It not only depends on whom you ask, but also when you ask. If the person is if it's in the morning, or if someone is tired, if you sit in a dark room like microscopists often do, or if the sun is shining through the window, all this might change your judgment. And as tissue agnostics, we are offering software to introduce observer independence in the analysis of these slides, and also academically I'm working, I will show this in the next slide, on algorithms for next generation histopathology. But we have to always keep in mind that software can do a lot, like increase the contrast, remove the background, make spectral unmixing and so on. But the basic is always our experiment. And if the experiment doesn't work, don't expect the software can give you good data. And in that sense, I would like to point to this paper, which was published almost a year ago, almost on the day a year ago, where the authors analyzed 16 commercially antibodies for a specific protein. Doesn't matter now which protein that is. The point is, these are antibodies, and they checked and found that of those 16 commercially available antibodies, only 3 performed well, meaning specifically bound the target protein and without cross-reaction. And there were no papers published on that protein using these 3 antibodies. And now, fasten your seatbelt. But not one published study has used those. About 15 papers described experiments using an antibody that didn't even bind the key protein. And these 15 papers were cited, collectively cited more than 3,000 times. Whew. So this is the background on which I have been working over the past almost 30 years, including my PhD thesis. And as I said, I have a commercial leg, which is the company Tissue Agnostics. Feel free to visit us outside at the booth if you want to know about the products. But also still an academic leg. I'm working at Queensland University of Technology, and here we are specifically working on a project that is called the Virtual Histopathologist for Precision Cancer Diagnostics. This is mainly development of deep learning algorithms for analysis of histological samples. Just a word about QUT, a little bit of advertisement for my university and for Australia. It's based in the wonderful city of Brisbane in Queensland, and it's doing pretty well in the international ranking. I belong to the Faculty of Health, School of Biomedical Science. I'm physically based at the wonderful institute or building. It's the Translational Research Institute of Australia, where QUT, University of Queensland and Mater Research, Mater is a private hospital chain in Australia, collaborate on realizing translational research aspects. And QUT will also start a School of Medicine in 2028. So if any students are around and would be interested to come to Australia, think of QUT, please. The company Tissue-Gnostics, which I co-founded in 2003, is located in the city of Vienna in Austria. So to be clear, there are no kangaroos in Austria, okay? But they are in Australia. And we are still located in this beautiful building close to the city center. But today we have a global network of subsidiaries. Like outside at the booth is my colleague Bogdan from Tissue-Gnostics Romania. Here is my colleague Felipe from Tissue-Gnostics Latam division. Please reach out. And in order to achieve this goal of bringing the idea of flow cytometry with state-of-the-art algorithms to the microscopic slide, we have been collaborating with academic partners, not only in Australia in form of QUT, where I am based, but also in Europe. And I would like to mention these 7 consortia. Maybe you have heard of the Marie Curie Innovative Training Networks. This is a funding scheme from the European Union, where there are always several universities from Europe and industry partners like Tissue-Gnostics come together to work on a specific project. And the EU funds each of these projects with typically 3 to 6 million euro. And the projects to which we participated so far were 8path, which looked into computational pathology, INSEM with Technical University Aachen, where we looked on cell migration studies, Alcatraz, looking on the anaplastic lymphoma kinase as a project in translational oncology, with University of Cambridge, the calcium sensing receptor network, Helical with Trinity College in Dublin, big data analysis by machine learning. These 5 projects already finished. Ongoing are these 2, SSBB with the Erasmus Medical Center in Rotterdam in the Netherlands on the increasing problem of multi-drug resistance in bacteria, and Eradicate with Medical University Vienna on stem cells and cancer stem cells. So with the instruments and technology we develop partially in these academic research groups and then come into the commercial software, we can offer observer independent analysis like of this sample, which then shows us that here there are 15.8% of the cells. Each dot here represents a single cell, and you can create gates like you know it from flow cytometry. We can also look into spatial analysis. The term spatial biology is nowadays very popular. I would like to mention that we already did spatial biology 15 years ago. We didn't call it spatial biology, we called it contextual tissue cytometry. And with this I would like to summarize. As of end of last month, we have 3,450 peer-reviewed publications. You see from where in the world they come. And here you see how the increases of papers per year published using tissue image cytometry. This shows which fields are used. Number 1 is actually cancer research, followed by neuroscience and immunology, but it can be applied in all disciplines of science. And with this, I would like to acknowledge the contribution of our academic partners in Europe, in Australia. I would also like to mention at USP in Sao Paulo, we have Professor Henning Ulrich, one of our scientific advisors, and Karen Jacobs. And this relates actually to the panel discussion we had before the coffee break. And I don't have the time to go in detail, but I encourage you to look into the Africa Microscopy Initiative, which is an initiative funded by the Zuckerberg Foundation and Bill Gates Foundation. And they offer high-end imaging equipment to researchers all over Africa. And I think this could be a very good model also for Latin America. With this, thank you very much for your attention. Please visit us at our booth. I don't know, is there? And feel free to reach out to me on LinkedIn. Thank you very much.
Thank you, Rupert. So our next speaker is Emiliano Medei. Emiliano works at the Instituto de Biofísica Carlos Chagas in Universidad Federal de Rio de Janeiro, Brazil. And today he will tell us about his work at the Bioimaging Multi-User Unit in Senabio. So let's welcome Emiliano with a round of applause.
¿Aló? ¿Puedo hablar acá? ¿Puedo hablar en este? Sí. Okay. Thank you so much for the invitation, and it's a really honor to be here. And I'll be really honest for you that my heart is beating fast now, and so emotional for me because I was a student here in the University of Buenos Aires when I started my MD, just 2 blocks here in the CBC, so en el CBC, acá en la ciudad universitaria. So, and it's not easy for me to back home after more than 20 years out of Argentina working in Brazil. So thank you so much for the invitation, and it's really a pleasure to be here. And thank you to Fernanda Moll for inviting me, and Kildare, our director of our institute, also for this beautiful meeting. So I will introduce you about our National Center of Bioimaging. So that it's places in the Federal University of Rio de Janeiro. So the concept of this building was to put together not only a lot of equipment, but it's put to some mentality, how to study from the molecule, from the structure to the molecule, and the bioimaging from whole animals. So to do or to perform of this kind of roadmap, and not only for drug discovery, but also for investigate different disease or physiopathology disease or basic research only. And we have first one structural biology unit with several NMR spectrometers, and the last one was this 900 spectrometers, and all of these are working on 7/24 every day, and are open for all people around Brazil, but not only Brazil, and from South America. Several people came to Rio de Janeiro to use this kind of equipment. So, and the next step is to understand that, as I told you, the most important is not only to have the equipment, the people, the persons that are working in these facilities. And since 2012, we have the honor to invite Kurt Wuttrich, who was a Nobel laureate in chemistry in 2002, and he started to collaborate with us in the Senabio, and this was really interesting because not only visit the unit and the facility, but also he was advisor of the postdoctoral fellowship, and he was incorporated in our postgraduate program. And the second unit is the microscopy, obviously, that Kildare here is the most indicated people to talk about the microscopy. Here is the authority in microscopy in our unit, and now under the direction of Marcia Tia, who is here also. This unit have several microscopes, and you know much more like me in microscopy, but as you can see, you have some light microscopies, and also the optical tracer is just working and installing, and different other microscopy for structure and cell and tissue. And in electronic microscopy, we have a large number also of microscopy. We want to highlight these two microscopy. First, the super resolution that is well working in our facility also, and this Aurija also that was published years ago. This beautiful image reconstruction in our facility. So again, it's important. I will remark again the people that work in there, and we have a core of technologists and technicians that not only help with the machine, but also to think and help to the guy that need, the investigator that need to perform the experiment, which kind of technique is better for you to use at the time of the experiment. I think that this is the advantage that you have. So, and where we are going. So now, this was a dream years ago, but now it's not more a dream. Now it's really, and our unit have these both and cutting-edge machines working, and not only working, but I want to highlight the content of Kildare, Marcia, and all unit of microscopy, because not only install the equipment, but also given given different training, hand-on training, and not only for Brazil, but also for South America and from the region. So this is really important for us, and to create this atmosphere that facilitate this kind of advance in this technique. And finally, but not less important, the third facility is for small animals. So do you remember we start in structural molecules, after to cells and tissue, and now, and the third building, all of them are in the really, really close one of the other, okay? So I am talking about 2,000 square meters one to the other, so no more than 100 meters. So in this one, we have we are able to study from the isolated organ to the whole animals. The isolated organs is, I put something that maybe could be interesting, that is hosted just in my lab now, but we are working with this system. This is a homemade system performing some friend that I will talk about. And we are able, with the local field fluorescent microscopy, we are able, by look at the heart with a fluorophore, and we are able to perform this recording of the cardiac action potentials and cardiac and calcium transient simultaneously, really, really close places. It was possible by Ariel Escobar, that was an professor in UC Merced, and Ariel was in biophysics, and I can talk so because he passed away this last December, and Ariel was like a brother for me, like a big father for me, and he was the guy, you know, who have the lucky to know him, and and so it allows us to publish different works with this technique. And this year we were very lucky to put this in the cover of the Nature Cardiovascular Research. So regarding the small animals, it's important to highlight this machine that is an MRI, PET MRI, and 7 Tesla. This is both together. We have just the upgrade this year that is allow us not only to perform the anatomical places, but also to overlap and to identify where we have the function, the cardiac function, or at least the cardiac metabolic function that is possible to record with the PET. So with this MRI, as I told you, this is a multi-user and center. So with this machine, we do not only make an preclinical science, but also, and you can see, we have for biology different animals, and we were able to study in depth different structure. We have an in vivo also Ibis lumina. We have a luciferase system, for example, with tripamostigot, and this is important for Chagas disease, for example, but several other fluorescent or luminescent molecules could be used in this system. And echocardiography for different structure also. And the most important is the translational. We have a partnership with the IDOR Institute of Research and Teaching that allow us to perform this translational research between the SenaBio and human also. And finally, but not less important, also we want to show you that we have a strong spread science program in our SenaBio. This is really important for us to integrate the community with our center, not only at the SenaBio, but also outside. We have a big program about 200 kilometers from our university. We bring all of the several equipment, and we perform echocardiography with the students, with the public students. We perform a satellite video, and sometimes the guys from microscopy have the electronic microscopy recording online, and the guys in the school are really, really crazy. Wow, I don't believe that I can see just how you acquire and biological imaging. That is impossible for me in this region. Imagine to see in this region. And so the last initiative of our center that is one of the most important partners is the postgraduate program. It's a professional postgraduate program to training the new generation of master and PhD students in bioimaging. This is a beautiful initiative, and also Kildare, Marcia, they have a strong responsibility in this project that is ongoing now. So finally, this is our team, our director, Professor Adalberto Vieira, who is the director of our center, and all of the collaborators and professors that are working in this center. Thank you so much for the attention.
Thank you very much. Hello. Thank you very much, Emiliano. And our last speaker for this session is Fernanda Tovar Moll. Fernanda is the CEO of the DOR Institute of Research and Education in Brazil, and she will tell us about imaging in clinical and translational research at the DOR Institute. Welcome, Fernanda.
So thank you so much. I'd like to, first of all, thank you, Kildare, to thank a lot Leah and also Oliver and Kildare for the kind invitation, and also for the partnerships that we probably want to even strengthen more now. So maybe I will talk a little bit about the continuation of what Emiliano said. I will try to show a little bit the work that we have been doing at IDOR and try to give an overview of actually opportunities for collaboration in human research as well. So DOR Institute for Research and Education, it's a not-for-profit research institute that we created 15 years ago in Brazil. It started in Rio de Janeiro, but now we are in different states. It's linked to a hospital network, not only hospitals, but also clinical outpatients units in Brazil, around 80 hospitals now, 79 actually, spread through these different states. And we at IDOR are responsible for research and education integrated with this hospital network. We do have labs in Rio de Janeiro and São Paulo and Bahia, and I will show a little bit how we work, but the data can be collected anywhere. Everywhere we have this dot spot here, we have actually, for example, clinical trials going on in the different states. Those are our numbers. I will not go into detail. We have around 100 researchers working in basic and also clinic research. We do have a grad program and also a master program, so, and also postdoc opportunities. We do have a lot of students here, so maybe in the website you can find some opportunities. And we covered pretty much the majority of the areas of the field in science in terms of healthcare or medicine, but a lot of translational research. So a lot of things that probably can be in common or can be opportunities for collaborations with all of you. Of course, we all fight for funding, not only in the Brazilian agencies, but also we do have some funding that comes from abroad, Brazil as well. We do have a lot of collaborations, a lot of strange collaboration with the Federal University of Rio de Janeiro, Fiocruz, USP, but also with other universities in Europe and also in Europe. And we should and we must collaborate more with Latin America. So this is something that we really need to do. So we do have publications with more than 1,000 institutions. And if we can see here, the majority in the global north. So we need, and it's one of the reasons that we are here collaborating with LABI and also with Bridge. So this is a Wellcome Trust actually project that I'm honored to be part of that together with other researchers. And then we are going to be talking about that tomorrow in different sites. But the main purpose of Bridge is actually to talk about data governance and also collaboration and data sharing. And I think you're going to have the opportunity to hear from us in different. Ivane is going to present, and also we have a poster presented out there. At IDOR, we have also Agents for Innovation, where we do some collaborations with startups, and also to also foster the science entrepreneurship as well. We don't have only one lab for each researchers. We work with platforms and all integrated. And I will try to show a little bit about what we have at IDOR. So in Rio, for example, our lab is works a lot with stem cells. And here, as you can see, these are mini brains produced from the urine of patients. And they do a lot of experiments, not only neurons, but also cardiomyocytes and other things. Here in São Paulo, we do have a molecular and cellular biology lab that's all open for different researchers around Brazil and, of course, Latin America. We do have also a molecular pathology lab with all those equipments that probably you are familiar. This is one that's actually is a nanostream. So because we have a lot of patients and samples, so this is one example of the researchers that the biologists are doing with the pathologists in the lab as well. In Rio, we have a lab for samples, so a lot of fluid analysis, including a Simoa, where we are also validating in different with different cohorts, let's say. In the Northeast, in Salvador, we do have a lab that's more specialized in advanced cell therapy. So they do like CAR T cells, they do clinical trials phase 1, and they are accredited by the JC committee. So it's the European accreditation for advanced cell or advanced therapy over there. And for example, Bruno Solano just came back from Jennifer's Download lab at IGI. We have a partnership with her, with the lab actually. And Bruno is starting the first clinical trial, phase 1 in CRISPR for sickle cell anemia, developed by him and by Ross, that's from Jennifer's lab, and we are starting that in Salvador. So made in Brazil, that one. For clinical trials, we do a lot of clinical trials and actually run in different states. So nowadays we have around 275 clinical trials going on. And starting the majority, Latin America is the majority of the trials arrives are like phase 2 or 3. We lack actually possibilities of phase 1, but it's starting to change. And we do have around 20 clinical trials phase 1. With the COVID, a lot of vaccine trials, of course, and based on these clinical trials and clinical research, not only commercial, but also no-commercial trials that are run by our researchers, we do also have a biobank. So the biobank from tissues and also from samples helps us a lot with different researchers. These are the main sponsors for commercial clinical trials. And of course, that's the majority of them. They help us to actually receive fundings to really help with their non-commercial clinical trials. But we are very happy when we can do things like that and have the publications like in New England journals. And we always say is that our dream is to have Latin American in the first and the last authorships of all those trials. So talking about imaging, we do have an imaging platform. We have around 90 MRIs. It's 3 Tesla MRI and others 1.5. All those data is integrated, so we can actually collect all those data and analyze together. Ivan Ibramati is the guy that's responsible for this unit at IDOR. And we do perform around 81,000 MRIs per month. So, so that's a huge amount of data that's integrated, can actually help us to run different protocols and also different research questions. So this is just an example of our unit in Rio. From the unit in Rio, Ivane can access the data that has been collected in the Northeast, for example. And there also we have a neuromodulation unit. And of course, post-processing and software development, those are things that we have been working a couple of years in neuro and now also in other fields using AI. Rosana Rodrigues will show this tomorrow in another panel. And this is just an example of how things work. So, of course, MRI is a huge piece, and I am a radiologist, so I think that's a very important piece here. But imaging without the clinical data, without the samples, without those things integrated, we cannot actually answer any question. So here's just an example of cohorts that we have been running. So we do have samples, we do have a patch, we can do a tracking, we have the clinical data, and of course we have imaging in different here, so Alzheimer's, multiple sclerosis, and others. Just an example, so we just validated, because we had this cohort before from studying dementia, we could validate the first sample of the first test for p-tau in the blood of patients, available for research and also is available already for the patients over there. And also, here just an example, we have been working with quantitative neuroimaging. This is actually my field, so I work with brain connectivity, and we apply those techniques to understand brain development and also neurodegeneration to different disorders, and even applying to things that just arrived. So these are just an example of brain. You all know about long COVID, and long COVID, we studied this too. So those are areas that you have change in the white matter of patients that actually develop memory impairment, executive impairment, and we see those as well. Just to finish, so this is something that we have been trying to do at IDOR. We tried to do translational and clinical integration, let's say. We did that. This is an example with the Zika virus. So we studied the brain of the kids. We also, Stevens was the first one that actually showed that the neuron was infected by the virus at our lab in Rio. Fernando Bos and others studied the virus and in the genetics. And this integration is possible because we have partnerships with a lot of people, including Senavia. So just to close what Emiliano just said, I was also very lucky to be there during 10 years together with Emiliano and Kildare at the UFRJ. I'm not a professor there anymore, but I'm still a collaborator over there. And we have been trying to do that. So those are patients with callosal dygenesis. We discovered a path that's an alternative path in humans, but we had to prove that in animals. So this is mice image showing the same path that we proved to be there because we also did microscopy in the other unit. So thanks a lot. That's what I was trying to just show a little bit. And actually the main message is to call you all to collaborate more in Latin America. We can do big things together. Thank you so much for the attention.
Thank you. Thank you, Fernanda. And now we thank, of course, all the speakers for the great presentations, and we invite them up here to the stage for our 10 minutes questions. So we will take the questions from the audience, both from the auditorium as well as from our online attendees. Thank you very much. Questions from the audience?
Hi. Well, thank you for everybody for their presentations. I wanted to ask Rupert if it's available, the list of antibodies that you have been able to troubleshoot and validate it for these things, because there are many initiatives, and sometimes it would be good that all these efforts are put together to be available for everybody. Thank you.
So obviously that paper was not from our group. I'm just citing it. Indeed, we do get feedback from clients, and we are about to build a list of antibodies from different vendors where we get confirmation from our users that they work together with our instrument. But this list is in preparation. It's not ready yet.
I was in the SBBF in Brazil a couple of weeks ago, and there was a similar effort also there that was mentioned. It's a huge effort.
It is.
It has to be a joint effort.
Yeah, there are several actually groups or papers who are trying to do a standardization of antibodies. And as I said, I'm a cell biologist by training. I spent my first year of my PhD with a set of antibodies which didn't work actually. Yeah. And we tried to develop enormous complicated protocols with RNase inhibitors and whatsoever, where the staining took a week until all the antibodies were gone. Then my boss said, "Now buy another batch, we try once more," and it worked instantly. Yeah. So I know this from my own experience and history.
Thank you. Another question from the audience? I have a question for mainly for the directors of the facilities, which is if you do have programs or grant-like tools to allow the access of the equipment that you manage, with no fee, for example, or that allows mostly early career researchers to access your technologies?
Yeah. So we do have like we do help also the design of the experiments, and also we do have the cost. Of course, sometimes it's just not fee, but also just, for example, insumes. How samples, but we do have it's open, and just usually you access another researcher, then you can apply for the use of the equipment. But we do have also some grants for young investigators that are also in our web page, not only from Rio or they don't need to be actually linked to our institution. They can be from different, anywhere. And they just we just received 15 grantees from a program called Ciência Pioneira. So you can also look to that.
You know, we are a very small, center, but still, lucky to the grant that we got from Chan Zuckerberg, we have the chance to offer a round of 20 fellowships for we call first experience with advanced bioimaging tools. And the purpose of that grants were for a visit of 10 days to prove that any of the technology or techniques that we develop in the lab were useful to answer a question. So after that, the compromise was that we will work together in searching for deeper funding that enable us to do a more stable collaboration. And I'm happy to tell that the model is nice, works, and we succeed in a first row in the Imaging for All, in the Academia Latinoamericana de Ciencia, Unión Biolac, Company of Biology. And so far, I will say that I believe, or I can see some sustainability of that after, Chan Zuckerberg, because now we can, regenerate for more funding and make a new call for this first experience and going again to the system and searching for extra funding. So I see there an opportunity for us without basically having to compromise money from our institution, which is for a country like Uruguay very difficult.
Thank you. Yeah, well, we can also remember that there are the LABI programs to access facilities in the whole region, so that's also something to keep in mind. Another question from the audience?
Yeah, for Lionel. Great talk, Lionel. A question on... If I have understood correctly, you are constructing the fifth diver microscope.
You are what, sorry?
Constructing, you are
yes,
making
yes,
the
yes,
fifth
yeah,
diver microscope. It looks amazing, 2 photons, 3 photons, so really amazing. What I am, but this is the fifth, I suppose, prototype. So still, what is missing, or what is the reason why it is a prototype? I mean, why? Which parts are not yet for commercial?
One of the things that it always surprised me how the industry developed. Basically, as far as I concern, and speaking with head of the products in companies and things like that, I mean, you have to go to the companies and say, this is a fantastic microscope. You have to pitch here, you have to publish data, you have to collaborate with many people until a company get interested in a prototype and spend 5-10 years in engineering the microscope in order to make a business of it. I believe now for diver it's too late, because the patent from LABI is already 10 years old. So basically, there will not be any company interested in pursuing an engineering that will basically give you a few years of revenues. So we are now establishing a startup in Uruguay, developing diver as a technology, not for the diver per se, but rather for the application of diver to clinics. So I don't have a straight answer, Jose Maria, unfortunately.
No, it's a very good answer.
But unfortunately, I see many great technologies here and there that if you are not the kind of person, and Enrico Graton, the person who developed this technology, never was a person pursuing, basically the technology for the purpose of commercializing. He was always open to share with anybody the technology, but not for the business perspective. And I knew that they have the chance to develop this diver with ISS 10 years ago, something like that. They didn't succeed with raising funding for prototyping and engineering, and it never happened. So a fantastic microscope. I completely convinced this is a fantastic microscope. There is no other two-photon microscope with the capabilities that diver can perform. Will never be commercialized.
Thank you.
Can I comment on this? I would like to comment on this as well, because I went through this myself as a scientist who made a company. And I think most of the scientists, and I speak from myself, lack a proper understanding of what it means to bring a scientific invention, a prototype, to a product. So I had my PhD finished, the prototype was ready, and I thought, great, we have a product, let's make a company. But the step from having a paper published or a prototype ready to have a product is gigantic. Yeah. This is not just the next step and then you're done. It took us years. You have to get the financing in place. You have to, especially when you go into clinics, all the quality management and regulatory aspects need to be met. Yeah. A paper is nice, but it's not enough. No one will give you a certificate to use this technology in a clinical setup for clinical tests or implementation studies, which might cost hundreds of thousands euro, US dollar, whatever, or even millions, and take many years. But this you don't need to do if you want to stay in research. But then the research market is much smaller, and companies might be less interested in picking this up. Then when they know they have something which they can sell to potentially every hospital. It's a very like walking through the desert, I would say.
Yeah, thank you very much, Rupert. The last one, let's comment.
No, is that this opened. I also started the company 15 years ago, and it went all the way through the normal internationalization and selling. But at some time, it would be interesting to know which is the venture capital market in South America. It would be very interesting to know. I have no idea. I know a little bit, and in Spain it was not so easy. So
thank you very much for that insight from industry. It's always good to know the different sides, right? So we're running out of time. Thank you very much for this amazing session, and we can continue the discussion in the coffee break.
Sentado ou em pé?
Just a picture here.
Okay.
So good morning, everyone. For this session, the speakers will have 5 minutes to show us projects and initiatives that are important to Latin America bioimaging community. We will have maybe 2 minutes to make questions or comments. So I would like to encourage you all to contact them directly, because every session is networking session if you will want. So to begin, I would like to welcome Charlotte Buckley from Universidade Maior Santiago, Chile.
Hi everyone, it's really great to be here today. I recently moved from Scotland to Chile, so this is such a fantastic introduction to the vibrant Latin American bioimaging community. So this one? Sorry, which one? Pardon. Sorry. Today I'm going to talk to you about some core techniques that I use in my research, which is calcium imaging and signal analysis. So I use this to try and understand intercellular signaling in blood vessels. So some of the main ways that cells release calcium from the internal stores are via IP3 receptor binding of IP3. So the ways that I can experimentally stimulate this are to either use an external agonist, which will bind to a GPCR on the surface of the cell and cause PLC to activate the breakdown of PIP2 into IP3. The other way that I can use is to use a caged compound, so a caged form of IP3. This is inactive until you shine UV light on cells where this compound has been incubated. And then you get release of the caging group, allowing you to really, really precisely release calcium in specific cells or within that cell. I image the calcium using Cal520. So I work with endothelial cells, and we really like the Cal520 indicator dye. And I use this either on freshly dissected primary arteries, which I dissect out and pin open and stain, or on primary endothelial cell patches, as you can see on the bottom here. The imaging systems that I use are fairly simple, actually. For the arterial preparations, I use an upright microscope, and for the cells, I use the inverted system. For each of these, we use a high sensitivity EMC-CD camera and LED illumination. And we like to keep the light levels really low, so often with neutral density filters in the way to keep the light levels very low. We design and 3D print sample holders. This is so that we can get nice laminar flow across the preparations, and also bring in micromanipulators and pipettes so that we can apply agonists in very specific locations on the preparations. And we use UV illumination to actually cause the photolysis. So these are wrap electronic systems. We use a laser illumination system, which allows you to really specifically choose the cells you want to shine the UV light on. Or here with the flash photolysis system, which is incredibly quick within a single 10 millisecond frame. So what can I do with these tools? So these are some of the different types of imaging that we can do. With the UV photolysis, which occurs within the central region here, you can see that once we get this uncaging, you get this beautiful calcium wave propagating out from the uncaging region. We can also specifically increase calcium in an IP3 independent manner here with MPEGTA, and then buffer calcium. So here I'm using the diazo-2 AM dye. Similarly, with agonist perfusion, here I'm applying acetylcholine, which is stimulating this IP3 generation within the cells. Always add in sulforhodamine to be able to tell exactly which cells the agonist is interacting with. An IP3 independent way of doing it is by putting on ionophores like ionomycin in this central video here. And then it can also buffer calcium with BAPTA, as you can see here. So lots of different ways, a quick overview of the different ways that we can actually stimulate calcium release and buffer. So to analyze this, we use a mixture of manual and automated analysis techniques. So manually labeling the cells, because we haven't been able to actually get any really good automated way of finding cell boundaries. So if anybody knows how to do that, please come chat to me. And then these locations and all of the stacks are loaded into a home-written Python script where we find the raw signals, identify the baseline, and find the F over F naught signals. To then identify the peaks, we take the derivative, and then with these peaks, we're able to identify active cells and extract loads of different parameters, such as the peak signal, time of signal, different number of oscillations. And then with these, we can then find the averages across different biological replicates. So with these techniques, we can look at thousands of cells from the same preparation and analyze the signals really rapidly. And I would say that this, all of this software is being put together into with a nice user interface. It's going to be published soon and be open source. So that's called CalTracker, so look out for that soon. And if anyone's interested in any of these techniques, please come see me at my poster. Thanks very much.
Thank you so much. Some comments or questions?
Thank you, Charlotte, for your amazing presentation. I would like to ask you if, considering the speed limit of your technique, if you can use this to analyze, for example, in cardiomyocytes and other, electrostimulate cells, if you have tested this. Thank you.
I haven't tested it, but yeah, I think that would be very doable, yeah.
Okay.
Yeah, I'd be happy to chat to you later about it.
Someone else?
Thank you, awesome presentation. I would like to know what kind of fluorescent proofs are you using? Because I'm also analyzing calcium currents, but we're contending with the photophysic property of the fluorescent proof about the making more bigger, like this trace in the calcium, like the channel opening. Mm-hmm. Like we have this deformation on the cynetic or the response of the calcium, because sometimes the photophysics or even the properties of the fluorophore are like shaping more this type of curve, like even the natural phenomena. And I would like to know, like, if you're contending with this kind of phenomena, or it's just irrelevant because of the temporal scales you're extracting.
So we use lots of different ways of releasing calcium. Obviously, if you're doing it IP3 dependent, IP3 independent, you get these different transients. Mm-hmm. We find quite different signals, you're right, when you release the caged IP3, especially if you're saturating it. But we find quite similar responses in terms of, like, with the ACH and the IP3, caged IP3. I wouldn't say we get, like, vastly different, averages at the end, but I'll have a think about it and come back to you.
Thank you. Thanks so much.
Okay, thanks very much.
Just an unlikely comment in this case. These sessions, they're not for questions. The idea is that you can go and visit their poster and you can ask the people there. No? So we can move forward on the next speakers and presentations, and you can attend the poster and ask them. Okay?
Now, please welcome Moara Lemos from Senabi, Universidade Federal do Rio de Janeiro. Ah, sim. Não, é Moara.
Hello everyone, I'm Moara. I'm a professor at the Senabio that Emiliano just introduced a few minutes ago. And today I'm going to talk about the sample preparation for cryo-electron tomography and lamella preparation under the perspective of flagellar structure in Trypanosoma cruzi by cryo-electron tomography. So Trypanosoma cruzi is the etiological agent of the Chagas disease, a disease that affects more than 7 million of people, mainly in Latin America. This protozoan parasite poses a single flagellum, which is persistent along of the cell cycle and has an impact on the cell motility and cell morphogenesis. The flagellum, it's a complex structure formed by the axoneme, which is composed by 9 microtubule doublets and the central pair, and by an extra axonomal structure named the paraflagellar rod. So we are interested, our group is interested in to study the intraflagellar transport, which is a bidirectional movement of flagellar proteins between in the matrix of the flagellum. So this transport is driven in the anterograde direction, bringing proteins from the cytoplasm to the tip, and this is driven by the kinesin. And in the tip, the cargo is made free, and then the dynein recycle the proteins that are in the tip from the cytoplasm. And then we can see these IFT trains, what is called this arrangement of proteins, as electron densities along in the side of the flagellum close to the specific doublets. Then it was the first observed in Trypanosoma cruzi by our group. Oops. Then we used the cryo-electron tomography strategies to study, to have a deep look into this intraflagellar transport. So we used 2 different strategies. So one was the lamella preparation to reach a deeper region of the cell body of the parasites. And then we could see this region, which is the base of the flagellum, where we can see these intraflagellar trains being formed as a small arrangements in zigzag in here showed by the arrows. And then along the flagellum between the axoneme and the flagellar membrane, we see longer trains sandwiched between these, the flagellar membrane and the axoneme. So when we look directly in the flagellum, because the thickness of the flagellum is thin enough to be directly observed under the microscope, we could see novel circular structures in the Trypanosoma cruzi flagellum, which is also located between the flagellar membrane and the axoneme. Those are organized as few structures pulled together, or in many circular structures forming like a big group, forming some kind of protrusions between the membrane of the flagellum. We also observed those thin filaments, very similar to actin filaments, observed in the tip of the flagellum. So our plans, those are our initial data coming from the recent microscope that we have, the Titan Cryos, and then we want to look to the molecular identity of those structures and compare them with the IFT trains. So if you're interested in cryo-electron tomography, sample preparation, trypanosomes, or intraflagellar transport, come and visit my poster, the number 30. So thank you very much.
And now the next speaker is Laura Estrada, the host from Universidade de Buenos Aires.
I wasn't prepared. It wasn't my turn. They couldn't find my slides. Okay, good, thank you. Okay, so first of all, I would like to thank LABI committee for for organizing this amazing workshop. It's always nice to have the opportunity to share with experiences with so many friends, colleagues, and students. So thank you, Oliver, Leah, and the entire team. So I'm Laura Estrada, I'm professor here. I'm the director of the Biophotonic group at the Quantum Electronic Laboratory at the School of Science. Our lab is right crossing the street, maybe in that direction, that direction, okay, good. So it's working, no? Okay. Okay, so in my group, we use infrared pulse lasers as tools to address biological questions. And the main interest of our group is we can divide it into two main categories. One is to study intracellular dynamics, protein transport, protein-protein interaction, protein-membrane interaction, protein oligomerization. And on the other hand, we use infrared lasers as a tool to fabricate optical fabrication of micro devices. So on the side of biological intracellular dynamics, we perform, do we have a laser here? Yes. We perform single particle tracking, so we can track proteins and molecules once at a time. And we also do 3D reconstruction of moving fibers inside living cells by orbital modulation. And we also perform and applied a lot of fluorescence correlation spectroscopy techniques from the very basic point FCS to more advanced fluorescent FCS methods. And we develop also new application of those methods, most of them in collaboration with Enrico Graton and his team during the last years. And on the side of optical microfabrications, we have developed a method and using photosensitive resins along with 2-photon excitation lasers to fabricate rapid and low-cost micro devices. So this is our very first microstructure. It's a micro messy playing football here. And from this very first microstructure, we now can fabricate 2D, 3D, and even fluorescent microstructures within minutes and, I don't know, maybe $1 or $2 cost. So of course, all these projects are highly interdisciplinary. So you can see here my local current collaborators. They are all from different institutions. Okay, so they are all from different institutions, but they are all from biology departments. So I'm a physicist, and so this is really interesting. So most of my actual collaborations are here in this same building. And in order to address these questions, we develop specific instrumentation. So this is how our homemade instruments looks like. So it's very different compared to a commercial microscope. So this is a 2-photon orbital tracking microscope. So this is not working, but okay. And we also develop our own software to acquire data and to analyze data. And regarding sample preparation, we have access to cell culture rooms and also clean rooms. So this is interesting because this is Natalia, Mariana. Mariana is a physicist, and he is Francisco, who is also a physicist. So we are able, with help of a lot of people, some of them are here, to do some wet lab biology work also. And finally, so this is the people, the students who joined my lab since I returned to Argentina, like from my postdoc with Enrico Graton, like 10 years ago, and they have done almost all the work in the poster. Some of them are still around, so you will probably be able to see Ignacio and Joaquín, María José and Constanza, Nico and Mariana, and I mean, please stop by our poster, and this is a nice opportunity to, it's an excuse, let's say, it's an excuse to chat, to discuss about these projects and other things you want to discuss with us. So please stop by, and probably we can also share a mate.
Thank you so much. Now I'm glad to welcome Greg Keating from Universidade Federal de Minas Gerais.
Okay, so I'm going to tell you about the spectroscopy SIMS network that we're building. I want to tell you a little background quickly. So I'm from Minas Gerais, Belo Horizonte, UFMG. You can see we have, so we have somewhere marked here, you can see the arrow. And this is where the center is located. We have a very specially, a building that's rather large. So we have 3 transmission scopes and 7 or 8 working scanning electron microscopes, all with different detectors. And we do lots of teaching and classes also in the center. You can see a couple of the microscopes here. But the point I want to go, and this is when I want to ask this question, what is your dream microscope? So Lionel has hundreds of dreams, sometimes nightmares. So I think all of us have gone through this phase where you've been around a couple of years and you all have the microscope that you want. And so most of those will be something where you have resolution in the nanometer range so that you can study cellular architecture at a high level and detect changes when you compare diseases and other methods. So that's a pretty good basic goal. But then there are also, you want something that you can directly detect and identify material that's present, proteins, lipids, carbohydrates. We've been hearing about, we're limited by antibodies that we have that don't work. And so most of you have probably done SDS page gels, so you know how many bands you have on a gel that are unknown. And so you sit there like, okay, what are these things that are important? And so these things have been running around in my mind. I have a lot of developmental biology questions that I still haven't been able to approach until now. So I retire next year, then I'm going back to the lab to work on those problems. So one of the first things I want to say is that I have 2 technologies, and I've wanted these for years, and now we have them. It's not a microscope. The first one is that you can see here is one of the volume EM techniques. Not spend too much time here. So you collect hundreds or thousands of sections in Harvard and I forget the institute in Seattle. They've collected over 6,000 serial sections, mounted those, and the connectome is all built on that basis. The thing that I want to remind you, if you look at this is one of our first, Mario is sitting here, he has a poster and he'll talk more about this. We have next year have a symposium or workshop over this. But the point I want to make here is that when you get this data, this is still in the initial phases of it. You can collect all this data and you get great images, but the big thing for me is you can do volumetric analysis. How much volume, how much area do you have for mitochondria? What does that change in Alzheimer's, in Parkinson's? So you can start doing, with any tissue you pick, so you can start looking at all sorts of morphometric data. Again, Mario has a poster over this. We can discuss more. The second point, the thing that I've always wanted for years, was some way to do mass spectrometry imaging, MSI. And so you can see, most of you, I'm presuming, will know most of these. So there are a couple of different ways that you ablate. You get ionization, material goes off, and then you measure it. And so those different methods of mass spectroscopy, now those are imaging. You can also image a tissue, but it's not very good. The resolution is not good, and you're never quite exactly knowing if you're in the right place. So now with these new systems, the newest systems that are coming, the secondary ion mass spectroscopy, they can't go into the way they do this, but they have now for biology new types of cluster beams that you can ablate, and you can get peptides and lipids and carbohydrates fairly intact, at least up to 20,000 Daltons. So let me jump here. So this is the second part, is that all of this came about a lot. We got a grant, but we didn't know anything about it. So then, because of CZI, I made a contact with this fellow, Sebastian Van Uffel, started talking to him. I won't go into details here, but then we met a Brazilian that's working in a very good lab in the UK, NPL, and he invited us to a workshop that happened to be in Brazil. So then we go to the workshop and we run into other people, and this is a fantastic group to be in a workshop, so we learned everything we did. We bought an instrument, I'll show you, but also there I met Lara Baque from Argentina. So started discussing with her the possibility of setting up a SIMS network. So now we've got these number of people, a lot of people scattered over the place. I don't have time to talk too much about any of the instruments they have. So we have a very good group and it's building. We have other people that are coming in from Europe. They want to really help us build this network. And so it will be our instrument will arrive in December and then probably be mounted in early next year. This is our instrument. So it's an Ion Optica. It didn't fit in our room, so we have to actually remodel the instrument because we can't change our room. Room is 6 meters by 5 meters, and it doesn't fit. So I, because of time, don't have time to go through this, but you can do also mass spec, and then you have all the peaks that you got. You can do this, but you can put all this together, and you can get imaging to do in 3D. You can do cutting down and do 3D imaging with this mass spectrometer. These again, we have a huge AI part of this project that's being constructed. We've already got people from our computer department are very interested, and also Google probably will help us. This is like, I just want to mention one or two little quick points here. So usually you have the multiplex that you do with all of these. This is in a case of 15 different multiplex. But when you use this TOF SIMS, there's a way, and people are using nanobodies, primary nanobodies, labeled with lanthanides. And so you can do up to, counting lanthanides and other metals, you can get 54 different biomarkers labeled with primary antibodies. Then you throw them all on at the same time, and you do one analysis, and you interpret the peaks. So instead of spending weeks, you spend one morning. And then you run the analysis, and so this is all coming out. So this is a professor from UFMG. She got one of the I4A grants that was mentioned earlier, so she's now spending a year in Sebastian's lab in Maastricht. So another connection that went further, and so now she's working on developing this and also analyzing all of the carbohydrates, and she has contact with INCA, the Brazilian Cancer Research Center. And I'll stop there. So anybody has questions?
The speakers also have a poster, so we can make questions and comments directly. Okay? Now the next speaker is Taina Avelino from CNPEM.
Hello, everyone. Thank you, LABI, for the invitation. Thank you, organizers, for the kindly invitation. Today I will talk a little bit about our experience in CNPEM with bioimaging. My focus will in optical microscopy. My name is Taina Avelino. I'm a bioimaging lead in CNPEM, specifically in LN Bio. CNPEM is a national research center located in Campinas, São Paulo, Brazil. CNPEM houses 4 national laboratories. One of them is devoted to nanotechnology, bioenergy, the Synchronous Light Source Laboratory, and the last one, Bioscience Laboratory, where we work with optical microscopy. In my work at LN Bio, we work to provide free of charge training and access to advanced optical microscopy we receive around of 100 of 15 per years of users. Mostly of them coming from Brazil, but we can receive users from across Latin America and the Caribbean. Monthly, we can offer grants for these users to come, do their experiments and receive training in optical microscopy. To know more, please visit my poster, number 39. A part of our work to spread the word of optical microscopy, we are working to developing methods across the scales. We are working with images. Oh, sorry. Spoiler. We are working with images from entire animals, from super resolutions. And we are trying to develop pipelines to correlate bioimaging from light microscopy to synchrotron imaging. We have a lot of beam lines in CNPen or synchrotron light source. So we are trying to combine all of our imaging modalities to resolve biological problems. To know more about this beautiful work, please visit Umberto's poster. Thank you, LABI. Thank you, all my friends, my group, and LABI Bio for all the support.
And now, Armando Castillo will talk about bioimaging in Panama.
Thank you. Sorry for the delay. My name is Armando Castillo from Panama. It's a pleasure to be here today. I'm going to explain a little bit of the story of the Center for Bioimaging in my institute. So, in the past, there was nothing. All the bioimaging capacities in Panama were located at Smithsonian Institute. As you know, Smithsonian Institute is an international mission located in Panama that do a lot of research there. The problem is that at any given moment, Smithsonian could deny us the use of their equipment. So basically, all the ecosystem of research in Panama depended from these capacities from Smithsonian. And this actually happened this year when they basically closed the bioimaging capacities, and also they had the shutdown, so we cannot access those resources. But we knew that from the past, so we basically apply for money from the Banco Interamericano de Desarrollo and acquired 2 new high-end equipment for bioimaging, this ESEM and this confocal microscope. And so we basically created our new bioimaging system in DICASAT. So we have these 2 equipment. We are expanding for more equipment in Panama. And we give services to the whole research community in Panama now. So we actually work with the fabricantes. We have this core facility. We give training to people in Panama, all sort of microscopy. We also produce our own in-house protocols, and we work very close with the researchers to give the best services we can. So one of the things we do is now we absorb all the capacities for bioimaging in Panama. So even Smithsonian is using our research centers now because they closed theirs. And we're basically working alongside all these different institutes in Panama giving services for bioimaging. So we now have like 18 different projects, 8 different research groups working with the confocal microscope, and a lot of research going on with, especially with biomaterials and semiconductors from different universities in Panama. So we joined LABI last year, thanks for a visit, surprise visit from Oliver. I think you came to Panama and say, hey, why you don't join? We are now part of Latin American Bioimaging. And this is the complex we work here. This is my institute in DICASAT here. And we are building a new building here. And we are buying new equipment. And one of those is high content, high throughput microscopy. We're also working with one of these 3. We have open convocatory for buying one of these equipment. We want to do a lot of research in cancer, and we are buying a cluster of supercomputing to work with AI, doing imaging as well in Panama. We already have different theses. We are about to publish this. Also, this is a student from Spain who came to Panama to actually do this with us. And we do a lot of research, publish outreach. So we push really hard with the media around so people can see the work we are doing in Panama. So we have a Instagram page, please follow us. And I'm going to show you really quick the work I do in Panama. I'm actually a neurophysiologist. I work with ants, and what I want to do is to work with ants and the evolution of their brains. So this is an antenna of an ant, and we actually study the brains of the ants and correlate the different sections of those brains with the sensory input. And with the students, we reconstruct these brains. We look at the different sections of these brains, and we compare a lot of species of ants to see how the brain evolves. So this is another ant. This is a nocturnal bee. This is a wasp. And one of the techniques we are trying to develop is how can we get through the cuticle without dissecting the brain. Easy. My student pretty much make them invisible. And we can now scan the whole brain inside without breaking the head. Really quick, then we reconstruct the whole thing, and we can do now whole months of the insects in one go. And we are applying this to other insects. This is an ant. We can see the whole muscles, all the structures, the brain, the glands, and without micro CT. And we are about to publish this as well. And we can apply the same technique to different insects. This is a mosquito here. And we can do all sorts of reconstructions in the brain. So if anyone wants to work with me on this kind of research, insects, small animals, we can do that here in Panama. And you are all invited to come to Panama to collaborate. And so some images we will get there. The team and the students.
Thank you, Armando. So, Oliver, do you have some announcements or? Okay, so thank you all. Let's networking, guys.
So thank you very much, all of the lightning talks. Really great job. So we invite you all to visit those posters and those work and talk also with the people. I really appreciate your work. Thank you very much. So we continue with the next session. The next session is the report of the working groups. As you know, and if you don't know, you must know that the working groups are an essential component of this network. It's a space where we really gather together and think strategically on how can we really address these regional challenges that we have. And we're going to bring a brief report of the working groups, and each of them then are going to be involved in different activities. The Training and Education Working Group is going to have one networking session here. Then the Data Science for Bioimaging Working Group, it's developing with Elixir a training on fair data management tomorrow. And the Outreach and Integration Working Group is going to have also a full activity on Wednesday that we hope also that you can participate in. So the first group, it's the Training and Education. Training and Education? El primero, Fer. Okay. But Training and Education Working Group, so please, if you can continue. It's 10 minutes. Yes. Victoria. Repeto. Marcela. Es el enlace.
Okay, hello everybody. We are the chairs from the working group Training and Education. Victoria, Marcela, Andres, and well, also Ivan was not able to come here to Argentina this time. So we're going to present what the working group has been doing this year. So next one. Oh no. Oh, it's yes. Yes. Okay. So the working group has been working for 4 years now. Yeah.
Yes, from the beginning. Yes.
From the beginning, and when we want to share these main goals that we have been working on these years. So one of the first one is to coordinate the activities in the region. So we began working with a survey to access to access the training opportunities in the region. So we found they were very dispersed, and many students from one country don't know the training opportunities from another country. So one of our first aims is just to be able to coordinate those activities. It's not working. Okay. Okay. No, yeah, thank you. All right. So the next goal is then take into account the available resources, realize that we could make our contribution there, improving the resources available, just with more courses on fundamentals, and Marcela and Andres are going to talk about that more. And also there was a need in the region to facilitate the exchange of experience among imaging scientists in Latin America, especially in their native languages, because, okay, we are here all talking in English, but sometimes students feel ashamed to speak in English. I would like to talk in their native languages, Portuguese and Spanish, so we make room for that also. And in the end, also to accelerate the application of imaging technologies in biomedical research through focus, training and exchange. So also the group has been helping to connect people to attend to apply for the grants, to travel from one facility to the other and to connect facilities. Okay, this is what we have been working. We have been mapping all the training opportunities in Latin America from the former years. And then from that, we began to work with the trainer initiatives. So Marcela?
Yes, in the first, the last slide was the beginning of this group, but in 2024, after the LABI meeting in Rio, we started to work together. And after this Train the Trainer event in Buenos Aires in the LABI Institute, we started to have our virtual meetings. This was the event that they joined together, and we started to talk with other users and microscopy trainers in Latin America. And after that, we decided, during our virtual meetings, we decided to start to work in two different subgroups, because we start to look at the main goals that we have, and we decided to start with two of these goals. One is to help and support the organization of fundamental in optical microscopy courses and also electrical microscopy courses. And we start with the fundamentals, and we decided to be partners, develop these courses in Uruguay and Argentina. And another work that we are now starting and we are conducting in the next year is our Curso Estrella de Microscopía. This is the name that one of our colleagues give to this course. And the idea is to give that, make a hybrid modality courses with virtual lessons and the practical sessions in each institution in person.
Oh.
Bueno, finish.
I'm sorry.
What?
Sorry.
Más despacio.
Thank you.
Okay, in April of this year, we performed the course of fundamental microscopy in Uruguay in the Institute Pasteur of Montevideo. Victoria and Andres comes to our unit, and we developed this course for local microscopy users. And after that, in August of this year, we also performed the Curso de Fundamentos in the LABI Institute in Buenos Aires, Argentina. Now we have a bigger team of excellent microscopy trainers from Argentina, from different parts of Argentina. And this is the other part of this subgroup of fundamentals. We are now working in generate this hybrid course with these lectures that is going to be online or recorded. We are talking in the group the way to do it. And the idea is that every institution or every university that is involved have a in-person practical activity in accordance with what are your available equipments in each region, in each country, to perform the practical lessons. And we are now working in this. Also, Ana Clara is give us a lot of help generating a complete new brand from the courses in Latin America. We have logo, we have the program and the flyers, and we are working with her. Thanks, Ana Clara, for all your help. And with the other,
okay,
this is the other objective.
Yes, and so as Marcela told you, we split into two groups because there was so much to do, and for just only one group. And the second group is the training education subgroup of exchange of experience that first we call IDEM because we wanted to make it in something that makes sense in Spanish and in Portuguese. So it's Intercambio de Experiencias en Microscopía, which makes sense also in Portuguese. So along with Ivan, we organized this series of, we are not calling seminars, but IDEMs, in which the main idea was like microscopy beneath the curtains. It means, okay, we are not going to give you a seminar about this wonderful paper and how it came, but we're going to talk about the things that go wrong and how we solve it finally to get the paper. Sample preparation, so the topics were like sample preparation beneath the scenes, so everything that goes wrong and how can you mend it to finally get your nice image. Involves fluorescent microscopy and electron microscopy, also experimental. Also experimental designs in Latin America. So when you have to face all these budget problems, how can we help each other, and cases that we're able to finally interact and go along with the experimental projects, and also how to design the working plan to make a short visit. So we get advice also about that. And we were very happy because we have more than 200 people registered from 10 different countries. And as I told you before, the seminars went both in Spanish and Portuguese, so everybody could participate in their own language. And also each session takes a short moment of presentation and a long moment of interactions using Menti, the online tool, so everybody just got put their ideas there. Okay. So having said that, thank you very much. And show the group is open, the working group is still open. You can show the LABI community, and in a few weeks we are going to give the final IDEM presentation, closing the year, because the public wants it more, so we're going to keep on talking about sample preparations. So we invite you to join and come to the virtual event. Thank you so much.
Shall I go? Yep. Okay, so hello everybody. My name is Adan Guerrero. I come from Mexico. I'm now representing the working group Data Science for Bioimaging. And so in this case, let me see if it will move. No, will won't. You are still changing the presentation? It is already there. Okay. Okay. So as you can see, the name here is quite different. Initially, it was called Image Data Science, and you can refer to whatever name you want, specifically because we want to solve problems, not naming things. And so this working group came out the last meeting in Rio de Janeiro. So here we see that logo previously. And indeed, we have a in-person session when we meet with all the people that were interested at that time. And we start asking questions in both English, Spanish, Portuguese, and whatever way we can find to communicate each other. And some of these objectives came out of that session. So this is a very young working group. It started 1 year ago. The idea started to materialize 1 year ago. And it was born with 4 different objectives. And all of them are correlated to a single objective, which is let's to help us each other to live and survive in this world of data science. And specifically, we identified that we need education, we need improve access to infrastructure, we need to promote the standards of quality internationally, and of course, we know, advocate for funding. But this is what we really want to talk about, the problems that we have found. So in all of these cases, the image data, science, analysis, management, all of these words came out. And so these were the major ideas. We need funding to develop infrastructure. It's hard to keep track of data and its availability. We need standards and guidelines for data storage and handling. We need help for image analysis. We need to learn to do image analysis. It's difficult to find those learning tools that are tailored to our needs. And we also need to have better access to computing resources. Infrastructure is not well distributed. And there is a limitation for mobility all everywhere. But this was one of the most important things that we realized there, that in some of the countries, the general concept of that data management is not present. We know about data analysis, but not really data management. So we start thinking what are the major ways to address these problems, and one of those is to identify avenues to promote long-term sustainability, like funding bodies, but also to increase capacities in training and education and so on and so forth. Okay, so and this is a very brief things that we are doing in Mexico. In Mexico, we found a neutral space for international collaboration, which is the International Center of Sciences, and we want to put there, we have there some courses of image analysis like this one of global bioimaging, but the thing that we want to do there in this is to create a node of bioimaging in Mexico, and this node will help, and it is not a hub, it is a node, will help to promote this initiative. So we have the space, we have the idea, but we don't have the money, but we are working for that. The main idea is to push forward these objectives, like pushing the increasing the capacities in education, promote network integration, and so on and so forth. Okay, but first, going forward, in the working group, we decide to survey all the community, not only start with ideas that we had in that meeting, but to ask you. So here we are, asking you to take your phone out, scan that QR code, and please help us with your help us to understand the needs that you have, because then we might come out with some solutions that emerge from the community. Of course, we start the surveys, I think last month ago it was released, and we have some outcome of this survey. And for example, when we speak about limitations, you can answer these surveys in Spanish, Portuguese, or English. And so the color, the orange is about highest limitation, and the blue is the lowest limitation. And there are limitations everywhere, of course. But let's what we can do to solve these problems. And one of the things what we have found that is valuable for the community is that workshops are important. Whether they are local, regional, or international, we need to organize workshops. That is one way to address these problems. Okay, and an important part is that the materials can be either in Portuguese, Spanish, or English, depending in the communities. And how to sustain this kind of initiatives. So people think, we ask whether we need a degree in a university, whether people is more comfortable with organizing workshops, whether they prefer classes within a program, etc. And what we found is that everything might be useful, but also, so this is related to formation, but also we asked about sustainability, and what came out, I mean, this is 27 person, okay? So that's why we need you guys. Is that we need networks to support to increase the outreach, the impacts of these initiatives, and the train the trainers programs that we have seen in some initiatives happening right now are very, very valuable for the community. Okay, and so people now is getting used to lead with scripts. Okay, these things are changing. Now people feels a bit more comfortable or have been exposed to working with the scripts, but at the end we have to develop the full ecosystem. An important part is that the way in which people normally have learned is by themselves, okay? And I mean, that is a way, but we need more formation. And one thing that came out is that it's not about having professional formation, like a degree. It can be more workshops, because we want to solve the problems right now, not in the future. So in the case of image analysis, workshops might come as a general solution. But this, I think that we were finishing analyzing this data today. The most important thing is in which language you want to prefer to receive the information. It's your own language, okay? So, and this is something that that we want to give it to you. You can put the materials maybe in English, but the courses have to be in your own language to reach a deeper understanding of what you are organizing. So this is it. If you didn't got the QR code, get it again and help us to continue with this.
Thank you very much, Adan, and also the rest of the group. The part are here. Fefo is on the back. So now we move to the Outreach and Integration Working Group, represented in this case Hernán Greco, Agustín Corbat, and Aide Hernández.
Good afternoon. Hi, my name is Hernán Greco. I'm part of the Outreach and Integration Group. The group is held by Mariana Denise, who was not able to come here due to some visa issues. She's a Mexican living in the US. Myself, Liciel Paulas, she's from Bolivia, and she's currently doing a PhD in the UK, right? And Aide Hernández, Mexican, who is around here, and she's also the winner of the T-shirt. Yeah. And we have Agustín Corbat, who is helping us as a local organizer of the following activities. So, as was already mentioned twice today, the training groups, working groups are an essential part of LABI. We have already heard about the training and education group, the data science for bioimaging, and here's the outreach and integration, which the purpose is to engage communities to inspire a next generation of scientists and connect our communities to address accessibility in microscopy. So it's like two goals, right? On one side is being able to connect with society. Why do we think microscopy is important? How do we connect the information and the knowledge about microscopy to those communities? And then also connect the local community of scientists which are scattered around the world in terms of microscopy, and in particular, which Latin Americans which are working currently in Latin America can connect to other Latin Americans working in the US, in Australia, in Europe, etc., to create some kind of bonds. And by the way, this afternoon we have the lecture from Carlos Bustamante. He's an excellent example of a person who has, by his academic career and degree, and he has worked a lot to sustain collaborations with local community in Latin America and to help labs in Latin America to develop by teaching here, by providing grants for students in Latin America to travel, by providing equipment. So we try to foster this type of connections because we understand that the diaspora of Latin Americans abroad can help a lot to increase our community. As I said, these are the 4 coordinators, right? And we have currently 17 group members of 8 different countries. And we have been doing some activities, meeting regularly, and basically trying to discuss what is the real nature. And what we realized immediately is that there is a big dispersion. I think it's very diverse, the continent, and also the outreach in the different parts of the world is very diverse. And so one of the, I would say that this was maybe the first activity that we had in the LABI Super Rest meeting that was held in Brazil. Right? It was a really nice activity with the community, going to the botanical garden, in which volunteers from LABI were engaging in different type of activities with people around. And what we realized here, and by connecting with other colleagues from all over the world, is that the reality in the outreach activities is very diverse. And a lot of people wanted to do outreach activities, but they didn't know how to start. You know, how should I organize an outreach activity? What topics should I include? What materials should I get? So we created this seminar series, which we did 4 of those, which was Ciencia en Conexiones. And the idea was to invite people doing outreach activities around the world and to share their thoughts, their knowledge, how they prepare, how they execute, and how they do the afterwards of an outreach activity. That was very useful, and we collected a lot of valuable information. These seminars are on the web, on YouTube, for you to read and to listen to. And the idea is that this, hopefully, it's already multiplying the amount of outreach activities that were discussed. And another thing that we realized that there is, in some countries, for example, last Saturday here in Argentina, there was a Noche de los Museos. There is a night which was very nice around the city, in which people were going to museums, including also to the School of Science, to connect with scientists and scientific activities. But this is not very common in all over the world, and we would like to help people start these type of things in their own places. There have been activities like this in the past. For example, some of you know a pint of science, for example, which is also a way in which people gather to get a beer, right, and discuss science, and that is very convenient. And so we are trying to help cities and colleagues and institutions to do this if they are not doing this already, right? And one thing that again comes is, okay, how do I start? Which type of information do I need? What can you share in terms of concrete activities? And that is why Aida is preparing a wonderful material that she would like to share with you. What's about?
Yes, so this manual of activities will be in Spanish, maybe in Portuguese. If someone from Brazil wants to be involved, just give me your name. So the main idea of the activities is to show the complete guideline about how to make an outreach activity. From what is the activity for, how many people do you need, which people will be involved, what is the topic that you need to know, and also what are you going to explain to the people. So actually we have only 4 activities from expansion microscopy, basic microscopy, and some sample preparation. And if you want to put your activities there, just send your email to LABI, and we will be put it online. So if you want to see also the material, we are creating the manual with all the design with Ana Clara. So in short time, it will be available in the LABI page.
Great, thanks. And then during this week, on Wednesday, we'll have 2 activities organized by LABI together with the School of Sciences. And so the first activity is being organized by Agustin. So Agustin, would you like to talk about it?
Yeah, okay. The first activity is more towards the integration part of the group. So the idea was to generate a space where students, undergrads, graduates, and also postdocs and researchers could share this space to start new collaborations, but also inspire the coming students and PhD students as well towards bioimaging, and also understanding that it's not completely mandatory to go to the global north to do your research, but there's plenty of collaborations available to do already in Latin America. So there's been a couple students that were asking, oh, I would like to travel abroad and do my PhD abroad or postdoc or even a stay, but they were a bit, they had this language barrier. And there's plenty of opportunities in Latin America, so we wanted to give this space for researchers from LABI to present their work to students from here to show them the possibilities they could go for.
Yeah, and the second activity that's going to happen simultaneously on Wednesday to the activity that Austin has just presented, it's about doing microscopy activities with high school and middle school teachers. Yeah, like around 25 years ago when I was finishing my undergrad thesis, they asked me to give a talk about microscopy, and I titled this talk, Microscopy is a point in which we get together, right? A punto de encuentro. And I really think about this, that microscopy is a great place to join different disciplines from physics, biology, chemistry, math, computer science, engineering, but also art, right? And nowadays in many countries in South America, they are trying to find projects in which to work in school. So instead of having the school organized by topics, they have projects that goes across this type of topics, right? And therefore, we thought that microscopy would be a great topic a great project to discuss with school teachers that they can bring to the lab. So what we are doing on Wednesday, around 25 school teachers are coming to the university, and we are going to use to do a project that was inspired by what Liciel has been doing in Bolivia, in which we get small lenses which are from laser pointers, and we combine that with a cell phone to look at different natural samples, but also technological samples, screens, microchips, etc. Right? And the idea is to show them how to build this type of microscope, how to use them, and then go deep into the samples to discuss about them, right? To put this in their own hands, and then the teachers are going to get a small bag with lenses so that they can repeat that with their own class in school. So we think that this has a multiplier effect, that it's not just what we are going to do with these 20 professors, but hopefully what they will multiply with their own students, right? So hopefully we will see also these things being applied. So this is going to happen on Wednesday, right? And so with that, I reached to an end. I invite you all to participate into the outreach and integration group. There are multiple things to do. It's a very diverse group. And we also think that the local realities are very different from country to country, from city to city, even within the same country. So if you like to start your own outreach activity and you don't know how, please send us an email. We'll be happy to participate and to know. And if you are doing an outreach activity and you want to share with us so that we can share with a larger community, please also do. Thank you very much.
So let's go to wrap up the day and see how we move forward today. No, yes, we can. Podemos cambiar. Yes. Thank you. Okay. So today, the day was focused, as you see, as you hear, and as you exchange, focus on technology in action, and that means showing and also hearing from successful cases of technology application, but also trying to exchange and to think strategically on sustainable models for technology adoption and innovation in imaging technologies to really reshape how we are doing the things, at least in Latin America, and integration with the rest of the ecosystem. As part of that work of exchange and technology adoption and implementation, and also an environment of exchange, for us the fellowship programs was something extremely strategic and valuable for us. And I want to show, as you already seen the last year with many cases, and in other presentations, if you have seen from LABI many cases, today I'm only focused on one case specifically, an interaction between Brazil and Chile in the light chain microscopy. In this case, we support Samuel Rocha from CAPI to travel to lithium in Chile. The thing here, of course, what we really think as valuable and to select those type of interactions, there was a highly advanced equipment in Brazil that was underutilized in terms of capacities and entrainment and training to really use at the maximum capacity that instrument, that equipment. So we support Samuel to travel to lithium at Chile. He trained there with Aníbal and all the team there in lithium that we have the lack of having here the three of them. And then 3 months later, he came back and not only taking the knowledge for himself, but giving a course for researchers on how to use that technology to really advance on using the light sheet as a tool for science. And that equipment now is producing more and more science. And that is the type of things that we are really working in LABI in terms of fellowships, and also how can really contribute to lasting collaborations, but at the same time, really contributing to accelerate the adoption of the technologies in Latin America. Because as we know, there are many things and many questions to answer before buying equipment. So, Aníbal, of course, and the ones that we already see, they are not unique cases. We are having really a lot of cases, and many of you are here in this room as part of these LABI fellows, and we are really happy also to having this opportunity to exchange in person. But of course, we are going to have a lot of stories more to tell now from this fellowship. So we really appreciate also your support and how you believe on this ecosystem and in this community. Thank you very much.